3. Evolution, a story without a mechanism. Matti Le Sola. Summary. At best, science is a search for truth about nature, how it functions and changes. At worst, it is a search for only naturalistic explanations for life's history. Is biology best explained by randomness or by a regulating intelligence? Can science prove one of the options to this philosophical question wrong? Several lines of experimental evidence show that novel functional genes and proteins cannot be formed de novo by chance processes. But can novel functional genes, proteins, or novel organisms be produced from existing ones by random methods? Individual genes, proteins, and microorganisms are easy to manipulate. They can be produced in large quantities and changed to the extreme in a laboratory, much more than could ever be happening in nature. Thus the laboratory experiments using random evolutionary methods are intelligently designed to study the limits of what randomness can do in biology, not what actually can happen in real life. The results of such experiments give a definite answer: there are narrow limits to the changes that random processes can achieve. They can never convert one gene to a basically different gene, one protein structure to a different structure, nor one microorganism to a different one. Thus evolution is a story without a mechanism, and adding the word theistic to it adds nothing to its explanatory power. At its best, science is a search for truth about nature, how it functions and changes. At worst, it is a search for only naturalistic explanations of life's history. Is biology best explained by randomness or by a regulating intelligence? The question goes back to Socrates. Can science provide an answer to this philosophical question? In Chapter 2, Stephen Meyer argues, based on experimental research, that the neo-Darwinian mechanism of mutation and selection does not provide an adequate explanation for the origin of genetic information. In this chapter, one will present several lines of experimental evidence to reinforce this argument, showing that novel functional genes and proteins cannot be formed de novo by chance processes. I will also ask whether novel functional genes, proteins, or novel organisms can be produced from existing ones by random methods. Individual genes, proteins, and microorganisms are easy to manipulate. They can be produced in large quantities and radically changed in the laboratory, much more than could ever happen in nature. Thus laboratory experiments that use random evolutionary methods to study the limits of what randomness can do in biology are themselves intelligently designed, not what actually can happen in real life. The results of such experiments give a definite answer: there are narrow limits to the changes that random processes can achieve. These experiments can never convert one gene to a basically different gene, one protein structure to a different structure, nor one microorganism to a different one. Thus evolution is a story without a mechanism, and adding the word theistic to it adds nothing to its explanatory power. Introduction. A team from my university has just won a gold medal in a synthetic biology competition. The presentation can be downloaded here, http://2015.igem.org. Team Aalto Helsinki. They produced propane using genetically engineered Escherichia coli bacteria. Throughout my entire career in universities and the biotech industry, my research teams have aimed at improving what nature can do, using both random and design methods. Improvement means tricking microbes or enzymes to do more than they naturally do, to produce compounds they naturally do not produce, to tolerate extreme conditions, etc. Ever since the discovery of microorganisms in the 19th century, they have been used to make a variety of products. Once a suitable production organism has been found, scientists try to increase production rates by random mutations, and, since the discovery of genes and genetic engineering tools, by systematic design. In this chapter, one want to point out, using some concrete examples, that both approaches have their limitations. Microorganisms and their proteins can change and be changed only within narrow limits. But before going into detail, it is important to define some terms. 2. Definition of terms. Philip Johnson and many others have pointed out that the term evolution has many meanings. Merriam-Webster defines evolution as a theory that the various types of animals and plants have their origin in other pre-existing types and that the distinguishable differences are due to modifications in successive generations, where, in line with the neo-Darwinian synthesis, modifications refer to changes in DNA produced by mutations and recombination. The changes to DNA occur at random and are fixed in successive generations by natural selection and/or genetic drift, processes studied by population genetics. Merriam-Webster defines design as deliberate purposive planning. According to this definition, milieu-type origin of life experiments are examples of design, as are all breeding experiments with plants and animals, no matter what the methods are. I want to emphasize that any experiment done by humans that tries to imitate what happens in nature is not what really happens in nature. For example, large-scale application of antibiotics in animal feeds has led to an increase in antibiotic-resistant bacteria. This is, however, not what happens naturally. It is a large-scale design experiment. The large-scale application of antibiotics is a man-made process that forces microorganisms to express antibiotic-resistant genes, or by mutation to modify their antibiotic receptors, nothing more. Every attempt to mimic evolution in the laboratory is an example of design, not an example of what actually happens in nature. All such experiments, as clever as they might be, search the limits of natural processes. Anything proceeding under artificial selection is not evolution but designed randomness experiments. What, then, happens in nature? Average mutation rates are in the order of 10 to 10, how often a certain base in DNA is mutated per cell division. Negative mutations exceed positive ones by 1,000 to 1,000,000-fold, according to various estimations. Jerry Bergman reviewed the topic of beneficial mutations. He did a simple internet literature search and found 453,732 hits for the word mutation, of which only 186 mentioned the word beneficial. In those 186 references, the presumed beneficial mutations consistently involved loss of function, loss of information, changes. In not a single case was new information created. A mutation, even if positive, must also be fixed in a population to be of any benefit. But the majority of mutations do not fix. They are lost. A lonely mutation in the big world will perish sooner or later. In real life all genomes degenerate, as shown using biologically relevant assumptions in numerical simulations of the mutation selection mechanism. All life has been degenerating since its first appearance. Life's direction is degeneration, not evolution. 3. Industrial microorganisms. Soon after their discovery, Louis Pasteur showed that microorganisms are not spontaneously formed under sterile conditions in a nutrient solution. In the light of the current understanding of the complexity of even the simplest microorganisms, such a possibility approaches impossibility. In 1887 Pasteur patented a method to produce beer and yeast. Soon all kinds of industries started to use isolated microorganisms, bacteria, yeasts, and fungi, in producing various chemicals, pharmaceuticals, and food products. The first examples were organic acids, acetone, butanol, and glycerol. During and after the Second World War, large-scale production of antibiotics began. In the 1950s and 1960s, production technologies were adapted to manufacture enzymes, amino acids, vitamins, and vaccines, and recently to produce diagnostic and therapeutic proteins. How does one improve a biotechnical production process? There are two basic ways: optimization of the process conditions, nutrients, starvation conditions, temperature, pH, oxygen availability, agitation speed, etc., and optimization of the production organism. Here I discuss only the latter. To improve the production of compound A, the first step is to go to nature and try to find microbial strains that produce more than the previously used one. Then, by random mutation techniques, the strain is made to overproduce the compound A in a protected laboratory setting, rendering the strain unable to survive in nature. If this is not enough, modern genetic techniques can be used to bring multiple copies of a specific gene into the strain or to design the whole production pathway into an organism. A. Random methods. Using random methods, impressive results have been obtained. Modern production organisms can produce hundreds of times more penicillin than the strain originally isolated by Alexander Fleming in 1929, or over 100 grams per liter of various acids, or tens of grams of enzymes secreted from the cell. Why does an organism overproduce a certain amino acid, for example? The reason is simple: its regulatory mechanism has been destroyed by a mutation. Nothing new has been created, rather, the existing regulatory pathway was made non-operational. Figure 3.1 shows schematically how this approach destroys the regulation of biosynthesis. Increased penicillin production by Penicillium chrysogenum is the result of increased copy numbers of relevant genes caused by classical strain improvement, mutation and selection. I take another example from my own experience. Xylitol is a 5-carbon sugar alcohol and a non-carriogenic, tooth-friendly sweetener. It is produced by chemical methods from hardwood species like birch. These trees have a fiber polymer called xylan, which is formed mainly of a long chain of xylose sugar molecules. Xylose can be converted to xylitol by reduction using candida. Yeasts. We wanted to improve the xylitol production process by inducing random mutations by toxic chemicals or UV light. Out of the thousands of mutants so created, we isolated a xylitol overproducing strain where the natural xylitol metabolism was retarded, illustrated in Figure 3.2. In nature such organisms are not viable, but they are useful as production organisms. In order not to lose such mutant organisms, they must be carefully kept isolated in aseptic conditions. B. Design by genetic engineering. Strains that overproduce a desired compound can also be designed by modern genetic engineering tools. In enzyme production, several copies of a specific gene can be added to the production strain. In the 1980s and 90s, the enzyme industry constructed new production strains using this method. Certain fungi like Aspergillus and Trichoderma, and bacteria like Bacillus subtilis, can secrete tens of grams of enzymes per liter of culture. Intracellular enzymes can also be overproduced. For example, more than 100 copies of xylose isomerase genes were introduced into Streptomyces rubiginosus in plasmids, extrachromosomal DNA. Such strains carry a large extra load of genes, which the organism does not need. Such an organism survives only as a pure culture in isolated reactors. In nature it would rapidly lose the extra genes. Genetic engineering tools have also been extensively used to bring new genes or even complete metabolic pathways into existing organisms. This is a difficult task, it takes a long time and has its limits. A real workhorse of biotechnology has been the Bacterium E. coli. It has been converted to produce such compounds as indigo blue, 1,3-propanediol for the chemical industry, isoprene as a starting material for synthetic rubber, human insulin, erythropoietin, and even fatty acids for biodiesel. These are examples of systematic design of reaction pathways. However, this is possible only when the basic metabolism of E. coli is not disturbed and the products are not toxic. Again, such organisms are not viable in nature and would quickly either die or lose the extra foreign genetic material. To demonstrate the limits of such processes, I again use xylitol as an example. We wanted to produce xylitol from glucose, which is both cheap and easily available. Baker's yeast is an efficient metabolizer of glucose and has a pathway to produce xylitol intermediates. Addition of two genes, see Figure 3.2, xylose reductase and xylitol dehydrogenase, into this organism should at least theoretically result in xylitol production. The idea worked, but the yield of xylitol was very low, only 2.5%. A similar approach with bacteria reached conversion yields of 23%, which is the highest reported for xylitol production from glucose with a bacterial strain. See Evolution experiments with microorganisms. All such experiments are examples of design, not processes that happen in nature. Once an organism is isolated and cultivated in a flask, its environment is no longer natural for the organism. Such experiments, however, demonstrate what the limits of natural changes are and, equally important, their direction. In 1968, Wu et al. Published results that showed that the Aerobacter aerogenes bacterium had learned to grow on xylitol. This was used as an example of evolution. What had actually happened? One mutation had destroyed the normal regulatory system in the bacteria. This resulted in a continuous production of one of its enzymes capable of reducing xylitol. Nothing new was created. Experiments carried out by Barry Hall shed more light on this. He studied E. coli and its ability to evolve. The bacterium can use the milk sugar lactose as its energy source. For this purpose it has a permease on its cell membrane to transport lactose into the cell and an enzyme called lactase, beta-galactosidase, to split lactose into two simple sugars, glucose and galactose, figure 3.3. Hall destroyed the lactase encoding gene, which resulted in mutants that could no longer use lactose as the energy source. When Hall cultivated these mutated cells in lactose-containing nutrient solution, mutants regularly appeared that could grow on lactose. What had happened? E. coli has an enzyme, EB, that closely resembles the lactase enzyme, though it is not able to degrade lactose. A single mutation in EB, the gene that codes for EB, is sufficient to allow slow growth on lactose. Only EB, the role of which is unclear, could be mutated to use lactose, although according to Hall, the best EB enzyme does not even approach the catalytic efficiency of the LacZ enzyme. A single mutation in a large bacterial population with billions of cells is well within the reach of random evolution. Richard Lenski's experiments with E. coli are probably the best-known long-term evolution simulating experiments. The meaning of these experiments has been thoroughly discussed by Michael Behe. The organism lost many of its capabilities, such as making flagella, and acquired nothing that it did not already have, such as citrate intake and utilization. In bacteria, evolution means losing genes. The species compete to grow and reproduce the fastest, and therefore get rid of all unnecessary functions. The most impressive evolutionary experiment so far reported was carried out by an international team using Salmonella enterica. In October 22, 2012, a report claimed that this was the first time anyone had demonstrated the origin of a new gene. In reality, a gene with a weak side activity was duplicated and the side activity was strengthened. Nothing more, nothing new. Yet this is how the work was described in the scientific literature. The emphasis is mine, to show at what points intelligence was introduced in the experiment. Researchers engineered a gene that governed the synthesis of the amino acid histidine, and also made some minor contributions to synthesizing another amino acid, tryptophan. They then placed multiple copies of the gene in Salmonella bacteria that did not have the normal gene for creating tryptophan. The Salmonella kept copying the beneficial effects of the gene making tryptophan, and over the course of 3,000 generations, the two functions diverged into two entirely different genes, marking the first time that researchers have directly observed the creation of an entirely new gene in a controlled laboratory setting. There is one more interesting evolution experiment carried out using E. coli. It is generally assumed that a multi-step mutational evolutionary path is possible if all the intermediary steps are functional and can be reached with a single mutation. The activity produced in this way may, however, be so weak that the cell must overexpress the hypothetical newly formed enzyme, in other words, produce large quantities of this enzyme. This is a huge strain to the cell because it has to use extra synthetic capacity for this. Therefore it is very well possible that the cell gets rid of such a weak activity even if it could be beneficial. Geiger et al. Studied what happened in such a case under laboratory conditions. They introduced two mutations into a TRPA gene that interfered with a tryptophan amino acid synthesis. One mutation destroyed the activity completely and the other one only partially. The cell could have regained the tryptophan synthesis pathway by two consecutive mutations. However, this did not happen. The experiment shows that, even if the cell could acquire a new activity by gene duplication and mutations, it would get rid of it, because the new activity is too large a burden. Although the described experiments are often promoted as evidence for evolution, they are actually all designed, and in fact they probe the limits of natural phenomena. They do not reflect what actually happens in nature. Recently, Chatterjee et al. Published an important paper. The authors evaluate the timescale that is needed for evolutionary innovations. Their key finding is that, for arriving at functional sequences largely different from the starting one, the processes take an exponentially long time, according to the sequence length required. When the search starts from a flat point in the fitness landscape, exactly the situation that exists when starting from a random sequence, for a typical sequence of 1,000 nucleotides, the search does not succeed, even if multiple populations are searching for the entire age of the Earth. The search is unsuccessful even when the target area is very broad. The estimated number of bacterial cells on Earth is about 1030. To give a specific example, let us assume that there are 1024 independent searches, each with population size n tilde106. The probability that at least one of those independent searches succeeds within 1014 generations for sequence length L equals 1000 and broad peak of C equals 1/2 is less than 10 to 26. The search is unsuccessful also when the targets are many and broad, regardless of what population size is modeled. This study thus supports the conclusion that there is a strict limit to what the commonly understood evolutionary processes can achieve. Tens of years of experimental evidence with microorganisms can be summarized as follows. Microorganisms can be designed to overproduce desired compounds by random mutations or by genetic engineering tools. Production of such organisms needs either huge mutation rates and systematic artificial selection or tedious construction and optimization of efficient pathways loaded into organisms, neither of which has resemblance in nature. Such organisms are not viable in natural conditions due to high mutational load or extra non-natural genetic load. Isolated microbial populations in laboratory experiments vary within narrow limits and lose information over time. 4. Proteins. There is general agreement among scientists that the sequence of building blocks of a biopolymer represents a type of molecularly coded information. It is the specific ordering of the nucleotides or amino acids building up DNA, RNA, or protein molecules that determine their structure and function. Proteins are the most versatile and efficient in terms of function. To be convinced of this, it is sufficient to take just a glance at a poster with metabolic pathways showing the plethora of reactions catalyzed by enzymes. 20 amino acids are the building blocks of the proteins present in all living organisms, from bacteria to humans. The average protein is about 300 amino acids in length, more precisely, 267 for bacterial and 361 for eukaryotic proteins. These 300 amino acids can be ordered in 20,300, 10,390 different ways. Scientists generally agree, based on several lines of experimental data, that more than one specific protein sequence is capable of performing a particular function. But scientists still debate the size of the fraction of functional protein molecules among non-functional ones, as well as how best to describe the functional information residing in proteins. The difficulty is confounded by experimental findings showing that there are protein families with more than 100,000 members having related but different sequences and, most likely, essentially the same structure and function. Moreover, proteins having different sequences and structures but similar functions are also known. How can one address this difficult issue? A. Rarity of functional proteins in protein space. Jack W. Szostak, Nobel laureate, 2009, and his co-workers have, over the last 20 years, made extensive studies related to evolution of proteins with new functions. Their work is of exceptional importance because of not only its methodological novelty but also its conceptual originality, depth, and breadth. Experimental studies with proteins became possible thanks to a major methodological breakthrough that enabled a mRNA molecule to remain covalently linked, after translation, to the protein whose sequence it encoded. Roberts and Szostak, Nemoto et al. One key advantage of this method is that researchers could start with a large population of different DNA molecules, similar in size to that used for selecting functional RNAs. The second major advantage is that the selection of functional proteins automatically results in the selection of their mRNA, allowing for cycles of reverse transcription, PCR amplification, transcription, translation, and new selection. Using this procedure, Keefe and Szostak examined the frequency of functional proteins among polypeptides whose amino acid sequences were almost random. The function selected for was binding to ATP, and the length of the proteins was 80 amino acids. 4 families of ATP-binding proteins were isolated from the starting library of 6 by 1012 sequences, indicating that roughly 1 in 1011 of the starting sequences possessed ATP-binding activity. According to the authors, in conclusion, we suggest that functional proteins are sufficiently common in protein sequence space, roughly 1 in 1011, that they may be discovered by entirely stochastic means, such as presumably operated when proteins were first used by living organisms. Kozulik and Laisola have recently made a careful analysis of these results and have concluded that, even with extremely conservative assumptions, the probability of finding ATP binding activity which would function in a cell would be less than 1 in 1032, which makes the formation of functional activity by random selection a practical impossibility. The 10 to 32 fraction conservatively derived is significantly higher than some earlier estimates. Thus Yockey, based on reported cytochrome C sequences, estimated that this fraction is 10 to 65. Reid-Haraldson and Sauer estimated that the fraction is 10 to 63. Later, Axe concluded from his studies with penicillin-degrading beta-lactamases that the probability of finding a functional enzyme among random sequences is about 10.77, 10.53. In a study of 4 large protein families, Durston and Chu estimated that functional sequences occupy an extremely small fraction of sequence space, in all cases lower than 10 to 100. On the other hand, the estimate of Taylor et al. Is that a library of 1024 members should contain an ROQ mutase. In view of these different figures, and given the paucity of experimental data at present, one can be sure that the fraction of functional proteins among random sequences is lower than 1 in 1020. Kozulik and Laisola are not the first ones to argue that there is a limit to what evolutionary processes can achieve. Behe, in The Edge of Evolution and in subsequent work, has provided strong evidence that such an edge exists. Sanford, in The Genetic Entropy, has explained not only why natural selection is poor at creating novelty in genomes, but also why it is incapable of preventing genome deterioration. Axe has given strong evidence as to why just a single new protein fold remains beyond the reach of evolutionary processes. B. Random modification of existing protein structures. Enzymes are widely used for many different applications, in washing powders, food manufacturing, the textile industry, animal feed, and chemical production, etc. Unfortunately, natural enzymes are not always suitable for industrial conditions where high temperatures, extremes of pH, and a variety of other chemicals interfere with enzymatic reactions. Using the tools of genetic engineering, it is possible to modify existing enzymes with various methods. One approach is to randomly mutate the gene coding for a given enzyme. The process is called directed evolution, which is an oxymoron, connecting 2 opposing terms. One then seeks to find better functioning mutants among the variants so created. Some amazing results have been achieved with this technique, Laisola and Turunen. Enzyme activity has been improved. Thermal and pH stability has increased. Specificity has changed. Side activities have improved. Stability against solvents and oxidants has improved. In spite of these achievements, the technique has its limitations. There must be a mutational pathway to the new structure. One must be able to create a large enough mutant library in order to find the rare positive mutants. One must have a rapid screening method to detect the rare positive mutants. Behe has estimated that the upper limit for a random mutational process is 2 to 3 simultaneous mutations in one protein. This is in harmony with Hall's results with lactose mutants. It also must be pointed out that directed evolution has nothing to do with what happens in nature. It is a name coined for a specific type of design experiments. Extremely high mutation rates, carefully chosen reaction conditions, use of tools of genetic engineering, selection of variants toward a desired goal, etc., are all hallmarks of design. See Design changes. I started to work with a xylanase enzyme in 1974, and since 1997 the aim of my research team was to modify its structure to improve its stability. We have designed so-called disulfide bridges into the molecule in order to stabilize it against extreme temperatures and influence its pH stability and profile, example, Xiong et al. Li et al. The bridges are formed automatically when 2 cysteine amino acids are in a correct position and distance with respect to each other. In Figure 3.4a is shown part of the xylanase gene, upper row, and the respective amino acids, lower row. The positions modified by genetic methods are shown in bold. In Figure 3.4b are shown the 2 positions where the gene has been mutated. The mutations lead to incorporation of 2 cysteines, C, codon TGC, to replace threonine, T, codon ACG, ACC. A disulfide bridge is formed spontaneously between the two cysteine residues in the model structure shown in Figure 3.5. The probability of forming one bridge randomly is very low, only 1 in 20 million. The probability of forming two bridges is as low as 1 in 4 by 1014 alternatives. In practice, this is completely out of reach for random methods, even further from what can actually happen in nature. Many research groups have tried to improve xylanase stability by random methods, and in some cases with good results, but have never created disulfide bridges. The probability of the formation of the above-described disulfide bridge is actually much lower because one has to change 5 nucleotides altogether in the 669 blocks long gene, which is one possibility in 1.4 by 1017. Behe's limit was 2 to 3 mutations. It has been suggested that new enzyme activities are formed so that the weak side activities are improved by random mutations. This is of course possible, but in such a case nothing new is formed, only the existing activity is strengthened, while the basic protein structure remains the same. Nothing evolves unless it already exists. A recent paper by Axe and Geiger emphasizes this point. Mutation and selection can improve good designs but can never invent a design. It has been suggested that new proteins are formed by duplication of genes. In such a case, the gene keeps its original function while the new copy is free to mutate to new functions. The starting point would be a functional protein, and it would not need to be built from scratch. This has, however, many problems. 1. Where did the original gene come from? 2. The duplicated gene can change. Only within narrow limits. 3. Over time, all genes degenerate. Sanford. Bloom et al. Have shown that extra stability allows more mutations and makes the protein structure more flexible without destroying its natural structure. This only emphasizes the point that no random process can change the basic structure of a given enzyme to a different structure. Geiger and Axe made a similar experiment, but chose two enzymes with structures which very closely resembled each other. They made 29 specific amino acid changes to one of the enzymes without being able to change its function to another one. They concluded that even this modest change would mean at least 7 nucleotide changes and, considering the known mutation rates, would take at least 1,027 years, which is longer than the estimated age of the Earth. This result and others like it challenge the conventional practice of inferring from similarity alone that transitions to new functions occurred by Darwinian evolution. Blanco et al. Studied experimentally the sequence space between two different small proteins having different folds. One was a 62 amino acid protein that folds as an 8-stranded orthogonal sheet sandwich, and another was a 57 amino acid protein that has a central helix packed against a 4-stranded sheet. The authors designed a gradual series of mutants in trying to understand whether there would be an evolutionary path from one fold to another. The conclusion of their study was that the sequence space between the two proteins is enormous. The results suggested that only a small fraction of this space would have adequate properties for folding into a unique structure. The sequence spaces of the two small proteins did not overlap, and a change from one fold to another could not be reached within a valid evolutionary trajectory. Thus it is much easier to build a new protein from scratch than to change a basic structure to a different one. I have now briefly reviewed the key results obtained during the last 3 decades of protein and especially enzyme engineering. The results can be summarized as follows. Proteins can be modified with random and specifically designed methods. But only within narrow limits. The changes are not fundamental, basic structures cannot be changed. All the experiments done are designed, and have searched much larger space than the natural processes could have searched. Even with huge amounts of intellectual input, nothing basically novel has been created. Knowledgeable intelligent agents would not try to change one protein structure to another by random experimentation, but would rather make a new one from the beginning. Why then? In 1972 I was sitting in the major lecture hall of the University of Helsinki as a young student of biochemistry. Francis Schaeffer had come to Helsinki to give lectures. During those lectures I realized how naive was my concept of truth. I bought all his books and started my reading in philosophy, which I previously thought to be of little or no value. Some years later I was reading, as part of my biochemistry studies, Albert Leininger's Biochemistry. The last chapter was about the origin of life, and I was surprised that, instead of solid science, it contained basically philosophical speculations. The random proteinoids synthesized by Sidney Fox were considered important in solving the problem of life's origin, and there was no discussion of the information problem. I also studied enzymes, by Dixon and Webb. They openly and honestly discussed the real problems in the origin of life speculations. While the Smith as the creation of life was an eye-opener, we have no idea where life and its huge information content came from. At that point I invited 3 of my professors to discuss these important issues with me. After 2 sessions I realized that they had no real answers but had swallowed what I will call the modern concept of truth. See discussion that follows, without any further consideration. One has to ask, why don't clear results and straightforward theoretical calculations convince scientists that evolution is only a story without a mechanism? The reason must be outside of science, it is in philosophy. In the Philebus of Plato, 429 to 347 BC, Socrates asked the key philosophical question: whether all this which they call the universe is left to the guidance of unreason and chance medley, or, on the contrary, as our fathers have declared, ordered and governed by a marvelous intelligence and wisdom. Only a few scientists openly express their philosophical starting point. The Nobel laureate Jack Szostak and his co-workers express with clarity their philosophical assumptions and conclusions. Thus, in a paper published in 2012, we read: simple chemistry in diverse environments on the early Earth led to the emergence of ever more complex chemistry and ultimately to the synthesis of the critical biological building blocks. At some point, the assembly of these materials into primitive cells enabled the emergence of Darwinian evolutionary behavior, followed by the gradual evolution of more complex life forms leading to modern life. What, then, is the modern concept of truth? After hundreds of discussions over the years, it is quite clear to me that very few natural scientists are aware of their philosophical commitments. Even fewer know about Friedrich Hegel and his influence on the concept of truth. This is how Schaeffer summarized Hegel's influence: all things are relativized. In so doing, Hegel changed the world. Kozulik and Laysola discussed to some extent the effect of Hegelian thinking on the acceptance of evolution. I have talked with hundreds of students and professors around the world over the past 45 years. The influence of Hegelian thinking, in association with naturalistic philosophy, is overwhelming. Clear contradictions are tolerated, trivial experimental results are interpreted to prove major philosophical concepts, and even when the experiments point in a different direction, they are interpreted as proving a presupposed point. Few scientists seem to be able to see the contradictions in the following statements. 1. The formation of building blocks explains the origin of buildings. 2. All biological change is evolution. 3. Information loss is information increase. 4. Similarity proves common ancestry. 5. Intelligently designed experiments are proofs of natural phenomena. The modern concept of truth means that even clear experimental results are either relativized or interpreted to mean just the opposite of what they actually show. Let us give these statements a closer look. 1 and 5. The origin of life experiments are intelligently designed laboratory experiments and at best produce, among large numbers of toxic chemicals, some of the many building blocks of life. This is extremely far from producing even the simplest living organism with its huge information content. Sometimes even the presence of water on the surface of planets is interpreted to be enough for life's origin. The same is true of the so-called directed evolution experiments of enzymes. They are considered to imitate Darwinian evolution, although they are only carefully designed laboratory experiments. 2 and 3. Observed biological changes only prove variation and adaptation. They are usually associated with information loss, not increase. 4. Similarity only shows similarity and does not explain the origin of similarity. 6. Historical background. This distinction between design and evolution was clear in 1859 when Charles Darwin published his Origin of Species. In 1844, 15 years before Darwin, Robert Chambers published Vestiges of the Natural History of Creation, suggesting that species transmutate under the guidance of a creative power, that is, by design. Chambers's idea was attacked strongly by the leading scientists of the time, including Thomas Huxley, as well as by the theologians of the day. Darwin explicitly excluded the possibility that natural selection operates under any direction or guidance. Just a few of many statements from 19th-century books show that the absolute exclusion of design, meaning the absence of guidance by intelligence, or teleology, was the major reason for the acceptance of Darwin's theory in his day. Another fact cannot fail to attract attention. When the theory of evolution was propounded in 1844 in the Vestiges of Creation, it was universally rejected. When proposed by Mr. Darwin, less than 20 years afterward, it was received with acclamation. Why is this? The facts are now what they were then. They were as well known then as they are now. The theory, so far as evolution is concerned, was then just what it is now. There is only one cause for the fact referred to, that we can think of. The Vestiges of Creation did not expressly or effectually exclude design. Darwin does. This is a reason assigned by the most zealous advocates of his theory for their adoption of it. But Mr. Darwin tells us that the use of the words plan of creation or unity of design are marks of ignorance. In other words, there is no plan of creation, there is no unity of design in the Darwinian hypothesis. The favorable point in the variation selected is never a design point. It is favorable simply through chance, or luck, or fortune, or accident, and it is selected by the hypothetical law, because a lucky chance has made it what it is. And further, Mr. Darwin stakes the main work of the development of life on natural selection. In effect, he says, give me a struggle for existence, give me the weak and strong taking part in that struggle, give me the weak going to the wall, and I do not require design to account for anything. That which struck the present writer most forcibly on his first perusal of The Origin of Species was the conviction that teleology, as commonly understood, had received its death blow at Mr. Darwin's hands. The curious thing in modern biology is the fact that all the evidences trying to prove the mechanism of evolution are actually examples of design. Darwin saw nothing in biology that could convince him of design, as evident from his correspondence with Asa Gray. Your question, what would convince me of design, is a poser. If I saw an angel come down to teach us good, and I was convinced from others seeing him that I was not mad, I should believe in design. If I could be convinced thoroughly that life and mind was in an unknown way a function of other imponderable force, I should be convinced. If man was made of brass or iron and no way connected with any other organism which had ever lived, I should perhaps be convinced. But this is childish writing. The confusion of the kind shown by many modern scientists started a long time ago, when some scientists and theologians, in order to harmonize their view of religion with Darwin's theory, essentially equated the meaning of the terms evolution and design. Here are quotations from two early theistic evolutionists. Transmutationism, an old name for the evolutionary hypothesis, which was conceived at one time to be the very antithesis to the two preceding conceptions, harmonizes well with them if the evolution be conceived to be orderly and designed. The question, therefore, is reduced to this: has the course of evolution been designed and guided, or not? In either case it is evolution, but if it has been guided it is creation also, and evolution is the method of creation. A modern theistic evolutionist, Kenneth Miller, says essentially the same thing. I see a planet bursting with evolutionary possibilities, a continuing creation in which the divine providence is manifest in every living thing. I see a science that tells us there is indeed a design to life. And the name of that design is evolution. For theistic evolutionists, terms like directed evolution, evolutionary strategy, evolutionary design, coordinated evolution, evolutionary creationism, theistic evolution, etc., make sense. Darwinists today tolerate these terms because that is the price they are willing to pay for maintaining the appearance of validity of the whole Darwinian dogma. When pressed about their true meaning, Darwinists usually say that such terms are mere metaphors. In a recent critical review article in Biocomplexity, David Snoke showed that the use of teleological concepts and terms is widespread in contemporary systems biology, and that therefore the scientists in that discipline are actually working under a design rather than an evolutionary program. Many have demanded that the intelligent design paradigm must come up with a successful, predictive, quantitative program for biology, but it seems that such a program already exists right under our noses. I agree with Snoke. I have discussed with Kozulik at length how, in biochemistry and molecular biology, an illusion of the continuation of the evolutionary research program is maintained, in significant measure, by using and accepting contradictions, contradictio in ejecto. If design is equated with evolution, there is no possibility for overthrowing evolution without also overthrowing design. My view is that nothing can be logically proven to people who, like Kenneth Miller, equate things that are contradictory. That two things cannot be both true and not true at the same time is a law, the principle of non-contradiction, well known by Plato and Aristotle. Now, one may try to argue that evolution and design are not contradictory. However, the writings of Charles Darwin and his contemporaries, and of many others ever since, prove that they are. Darwin and his followers explicitly claim that chance variation and the law of natural selection have created all species of living organisms. If so, design is ruled out. In contrast, if design is the cause of the creation of species, then chance and law are ruled out as the creators of species. 4. Are present proposals on chemical evolutionary mechanisms accurately pointing toward first life? James M. Tor. Summary. Abiogenesis is the prebiotic process wherein life, such as a cell, arises from non-living materials such as simple organic compounds. Long before evolution can even begin, the origin of first life, that first cell, would have to come from some simpler non-living molecules. On Earth, the essential molecules for life as we know it are carbohydrates, also called sugars or saccharides, nucleic acids, lipids, and proteins, polymers of amino acids. Described is the process by which organic synthesis is performed, and the considerations that are generally required to synthesize a complex system where many molecular parts come together to operate concertedly. This will be demonstrated in the synthesis of nanomachines. Then considered will be some proposals that others have espoused for the synthesis of carbohydrates and carbohydrate-bearing nucleotide bases, from a prebiotic milieu. Briefly mentioned will be the obstacles to the much more difficult task of having the molecular building blocks assemble into a functional system. Not considered are scientifically unknown entities that have been proposed to have seeded life on Earth, such as a design agent or panspermia. An opinion will be rendered showing that the strongest evidence against the proposals of current prebiotic research is the researchers' own data. The current proposals can retard the field from discovering the scientific solutions since they seem to be directing researchers down paths of futility. Any account of the origin of the first life must include a mechanism for the generation of the chemicals needed for life, and then for how life arose from those pre-existing non-living chemicals. Abiogenesis proposals attempt to explain how chemical processes transformed pre-existing non-living chemicals into more complex information-bearing molecules such as DNA, RNA, and proteins. For an account of the origin of life to be realistic, there must be chemical processes that can successfully arrange simple organic compounds into complex biologically relevant macromolecules and living cells. Life requires carbohydrates, nucleic acids, lipids, and proteins. But what is the chemistry behind their origin? What is the origin of metabolism, or of the information storage and processing systems that depend on these complex biochemical compounds? My experience working in synthetic chemistry, building relatively simple nanomachines, has led me to be skeptical of proposals for the origin of the requisite chemical building blocks necessary for life. Some biologists seem to think that there are well-understood prebiotic molecular mechanisms for the synthesis of carbohydrates or proteins, lipids or nucleic acids. They have been grossly misinformed. Others think that, if not yet known, such chemical pathways will soon be identified. To me, these biologists are naively optimistic. What they hope for will not happen anytime soon. And no wonder, few biologists have ever synthesized a complex molecule ab initio. My experience with organic synthesis leads me to suggest that chemistry acting on its own simply does not do what it would need to do to generate the biologically relevant macromolecules, let alone the complex nanosystems in a living cell. I'd like to explain the reasons for my skepticism in more detail. Designing molecules. Let's begin by discussing the process of molecular design and synthesis in general, what it takes to successfully build a molecule to perform a particular function. The initial design is important. Sometimes molecular designs are computer-assisted, but more often than not, the initial steps are done on paper. A target. must first be drawn or otherwise designated. This is no trivial task. In some cases, chemists have seen the target in a related system. In other cases, they guess the target's properties on the basis of its molecular weight, its shape, the moieties appended to the main backbone, and its functional capacities. Once a target is selected, retrosynthesis is next, whether on paper or on a computer screen. Placing the target at the top, the chemist draws an inverted tree, or graph, one step down at a time, into multiple branch points, until he reaches a level where starting materials are at hand. The inverted tree is then pruned. Certain branches lead to dead ends. They are lopped off. Further refinement of various routes leads to a set of desired paths. These are the routes that can be attempted in the laboratory. Given a target and a path to get there, the synthetic chemist must now try a number of chemical permutations. Each step may need to be optimized, and each step must be considered with respect to specific reaction site modifications and different reaction rates. What is desired is often ever so slightly different in structure from what is not. If product A is a mirror image of product B, one left-handed and the other right-handed, separation becomes a time-consuming and challenging task, one requiring complementary mirror image structures. Many molecules in natural biological systems are homochiral, meaning only left-handed or right-hand molecules are used, not both. Their mirror images cannot do their work. In addition, few reactions ever afford a 100% yield, few reactions are free of deleterious byproducts. Purification is essential. If byproducts are left in reaction, they result in complex mixtures that render further reactions impossible to execute correctly. After purification, a number of different spectroscopic and spectrometric methods must be used to confirm the resulting molecular structures. Make the wrong molecular intermediate, the synthetic chemist quickly learns, and all subsequent steps are compromised. Finally, intermediate products are often unstable in air, sunlight or room light, or in water. Synthetic chemists must work in seconds or minutes to prevent destructive natural processes or chemical reactions from taking over. Building nanovehicles. As an example of what it takes to synthesize organic compounds, consider the synthesis of a molecular machine, a nanovehicle, a simple unimolecular structure that can translate itself along a surface when supplied with thermal or photonic energy. My colleagues and I make these relatively simple machines in the laboratory, and what we have learned about synthesizing nanovehicles has been published in numerous peer-reviewed papers. We set out to design nanotrucks and nanocars that can move across gold surfaces. They consist of three basic molecular mechanical parts: fullerene wheels, a chassis made of fused aromatic rings or oligo, phenylene ethynylene, OPEs, and alkenyl axles. See Figure 4.1a and 1b for trucks and 1d and 1e for cars. Ours was the first molecular-sized machine that incorporated mechanical components, such as wheels and axles, with movement at the single molecular level. The rolling motion of these nanocars resembled the rolling motion of macroscopic cars. We built two different kinds of nanovehicles, a rigid structure in nanotrucks 1A and 1B, the precursors along the synthetic route to these compounds, such as 1C, and a semi-rigid structure in nanocars 1D and 1E. All these designs were necessary because we discovered as we went along that better flexibility of the chassis structure combined with the increased number of alkyl units dramatically increased the solubility of the fullerene-wheeled structures in the organic solvents in which they were synthesized. The process was not straightforward. First we had to work out a way to attach fullerene wheels, a 60-carbon sphere, to the alkenyl axles, then a way to build a chassis of the appropriate structure, and attach the axles to the chassis. New reactions under new conditions had to be worked out in each case. For the nanotrucks, the first and second structures proved unworkable due to stiffness of the chassis and insolubility. We then modified the chassis design. A semi-rigid Z-shaped chassis for nanocars remedied the difficulties. The first nanocar structure was still too insoluble to properly purify, but the nanocar 1E, the fourth overall design, finally could be adequately purified and characterized, i.e., its molecular structure could be determined, for further study. Notice that all this design and experimentation required considerable knowledge and skill. Yet even with all these efforts, the properties of each design could not be predicted a priori, and as problems were encountered, things had to be restarted and redesigned repeatedly. In addition, many reagents were purchased to use in this protocol, otherwise the protocol would need to include steps for their synthesis and purification as well. Pre-treatment of solvents was needed so that the system would not be contaminated by impurities such as oxygen, which retard or mitigate the desired reactions. Purification was required at each step since the chemistry rarely affords the chemist materials that are of sufficient purity for use in subsequent steps. Each product needed a different purification protocol. Nature would not have this luxury as it moves toward the molecules needed for first life, or that first living cell. With nanocar 1E, we have been able to demonstrate the action of fullerene wheel architecture at the single molecular level. Evidence for thermally induced wheel-assisted rolling motion in the nanocar 1E on gold was obtained by scanning tunneling microscopy, Figure 4.2. The work that I have just described is merely a brief sketch of the processes we went through to synthesize the nanocars. The details are far richer. So here's something to consider as we think about the problem of the origin of life, the molecular machines and the information processing system that cells use to synthesize macromolecules are far more complicated than anything illustrated here. Designing nanocars is child's play in comparison to the complex molecular machinery and information processing systems at work in the synthesis of proteins, enzymes, DNA, RNA, and polysaccharides, let alone their assembly into complex functional macroscopic systems, a point that has become increasingly apparent to me as we've learned more about how difficult it is to build and improve our relatively simple nanocars. Cars with motors. Our first-generation vehicles operated without motors. We later made motorized versions that were more complex to design and synthesize. The motors are ultraviolet light active, they rotate unidirectionally. The first motorized versions that we made rotated at 1.8 revolutions per hour. Too slow. But with a redesign, which took us back to step 1 in the synthesis, we achieved a light-activated nanocar whose motor spins at 3 MHz, 3 million rotations per second, Fig. 4.3. Fast enough. The plan to synthesize the fast nanocar, Fig. 4.3f, involved a modular approach in which the coupling of the axles and the stator represented the last step. In the following indented paragraph, I present the description of the motor's synthesis in its more technical form so as to highlight the many steps and specialized conditions. According to Scheme 1, shown below, heating ketone 1 to reflux in an ethanol and hydrazine solution produced the rotor, hydrazone 2. The conversion of ketone 3 into Thi1,4 was improved by decreasing both the concentration and the reaction time from those in the published procedure. The generation of the sterically hindered double bond between the rotor and the stator utilized Barton-Kellogg coupling. Hydrazone 2 was oxidized to the unstable diazo intermediate 5 using manganese dioxide by careful temperature control. The inorganic residue was removed by filtration in a setup that enforced the strict exclusion of air, oxygen, and moisture. Thi1,4 was added to the deep purple filtrate. A 2+3 cycloaddition occurred and evolution of nitrogen gas indicated the formation of episulfide. 6. The white solid episulfide 6 was then treated with trimethyl phosphate in a screw-cap tube at 130 degrees Celsius to yield the molecular motor 7 as a mixture of isomers. Isomers are chemical structures that have the same order of attachment of atoms, but different three-dimensional structures. Notice all the places where the chemist was actively involved in carefully controlling reaction conditions. Scheme 1, above, illustrates the compounds and steps involved. We attempted the final assembly of the fast nanocar, Scheme 2, but coupling between TMSA and motor 7 did not afford the desired BIS-coupled product. A more reactive catalyst was used, but the result was disappointing because of a high degree of decomposition. By changing reagents and solvents again, a TIPS, protected BIS acetylene motor 8 was produced. The yield was excellent. Motor 8 had its TIPS groups removed, producing dialkyne 9 in quantitative yield. Dialkyne 9 and previously synthesized Axle 10 were then coupled to produce the fast nanocar in moderate yield, Scheme 2, Route I. Next, we tried a more convergent synthetic pathway, Scheme 2, Route 2, using coupling between motor 7 and alkynylated Axle 11, and applying conditions analogous to those for the synthesis of motor 8. The fast nanocar was thus obtained but in lower overall yield than that obtained from Route I. This underscores a common occurrence in organic synthesis. Even with modular approaches, small changes in the structure of the reactants make for enormous differences in reactivity. There is no simple workaround. Slow to fast. Consider the differences between motorized slow and fast nanocars, Fig. 4.3. A small change in the rotors had an enormous impact on the rate of their unidirectional rotation, 1.8 revolutions per hour for the slow nanocar and 3 million rotations per second for the fast nanocar. The rotor portion in the slow nanocar has a 6-membered ring bearing a sulfur atom, and the fast motor nanocar has a 5-membered ring bearing all carbons. What was involved in going from the slow to the fast nanocar? We had to start with keton 12, see figure 4.5, for the slow motor, and then use keton 1, see figure 4.5, for the fast motor. But ketones 12 and 1 are derived from entirely different starting materials. There is no known method simply to expunge the sulfur atom in motor 12 and obtain keton 1. It is all very easy on the blackboard, of course. One can simply erase atoms at will and show keton 12 becoming keton 1, figure 4.5. But chemistry does not happen that way. Even the oxidative expulsion of SO2 from keton 12 by the Ramberg-Backlund method would not work, since the sulfur is bound aromatically. We could have left keton 12 in a flask for millions of years and it would not form keton 1 by any known or rational thermal, reductive, photochemical, or enzymatic method. This is not unusual when related compounds have clearly different starting points in organic chemistry. It is typical. Wheel changes. Why did we change the fullerene wheels from nanocars in figure 4.1 to the carborane-wheeled motorized nanocars in figures 4.3e and 4.3f? Because we had to. There is no way to achieve motor functionality in motorized nanocars using fullerene wheels. We did not know that until we had already built motorized nanocars with fullerene wheels. We learned that after the fact. To our disappointment, when the motors are photoexcited, they immediately transfer their energy to the fullerene wheels so that the motors do not rotate. Thus we had to change the wheels on the motorized cars to carboranes, since carboranes do not accept energy from the photo-excited motors. The carborane wheels were not suitable for use on metal due to their low adhesion. But the carborane wheels worked well on glass. We have used them extensively to study nanocar motion on glass surfaces. A key point: parts are not always easily interchangeable without severe and unexpected consequences. When working environments change, drastic changes in molecular structure are often required to retain the system's functions. Here is a summary of the process that led to functional nanocars, including further details about difficulties encountered. Determine a target? Done. Molecular machines. Insurmountable problems? Yes. The first generation of nanotrucks had insurmountable solubility problems. Although organic chemists have at least 50 widely differing solvents and solvent polarities from which to choose, we could find no satisfactory solution. Start over? Yes, of course. Redesign? Done. Molecular flexibility, specifically a less rigid chassis, was needed, nanocars 1B to 1D. When we added a motor to the nanocars, the former chassis proved insufficient. We had to redesign the intermediates in order to affix the motors. Conveniently, we could store all new intermediates in the freezer to prevent their decomposition while we built the motors, and were able to access the published work of the Feringa group from the Netherlands in order to build upon their initial designs. But even then, starting at step 1 for the motors, we had to build in functional handles that would let them be accepted into the chassis and wheel assemblies. Only after constructing the new motors did we learn that they were incompatible with the well-developed fullerene wheels due to energy transfer problems. Redesign? Yes. When we desired to go from a slow motor to a fast motor, though the stator was reusable, the rotor was not. The rotor had to be redesigned, from step 1, so as to become a faster unidirectional rotator. And again? Of course. Once we changed the wheels to make them suitable to work with the motors, we learned that the new wheels were not compatible with a gold surface. So we had to change to a glass surface. The glass surface was not compatible with our imaging technique by STM, which requires a conductive surface. Glass is an insulator. Start from scratch? To image the nanocars on glass, we had to use a technique called single-molecule fluorescence microscopy, SMFM, but another problem arose: the nanocars were not fluorescent. This meant appending a fluorophore to the chassis. But we first had to resynthesize a new chassis to which the fluorophore appendage could be attached. We could have purchased a pre-synthesized fluorophore from a chemical company. These bulky fluorophores slowed the nanocars. So, starting from scratch again, we built a chassis with fluorescent axles. These we had to build using entirely different chemistry than we had used in the past cases. Sadly, motor energy transfer to the now fluorescent axles decreased the efficiency of the motors. Newer versions have motor excitation frequencies optically far separated from the fluorophore excitation frequencies. We know what frequencies to target because we can record the optical spectra using UV visible absorbance and emission spectroscopies. Yes, but even our optimized yields were not quantitative, 100%. First-time reaction yields were usually very low, sometimes as low as 0%. Only after repeated trials under different conditions could we attain a yield range of 50% to 60%, and sometimes not even that. Most organic chemists would agree that even with extensive planning, 90% of reactions are failures. Substrates and conditions must be repeatedly modified to secure respectable and usable yields. At each step a massive amount of time is spent on separations and optimizations. If byproducts are permitted to accumulate, they can consume the new step's reagents and alter the course of the reaction. After every 1 or 2 steps there must be purification. If all our reactions were near 100% yield, it would ease the separation problems. But this can take years to achieve, if it is possible at all. And even then, sufficient atom efficiency is very rare. Byproducts from the other reagents fill the system. High atom efficient reactions are even harder to achieve. The loss of materials is expensive. In most cases, these byproducts cannot be converted back to usable compounds in an efficient way. Developing a scrubber system for degrading these products back to usable starting materials would, in most cases, take more time and money than developing the original target synthetic routes themselves. The separated byproducts are put into waste disposal containers and sent for destruction through combustion. Unlike what would have been present in prebiotic times, we have plenteous resources available. We use petrochemicals as our major feedstocks, and these come in enormous amounts from fine chemicals producers. Large amounts of energy come from power grids. We have the convenience of ordering many of the requisite reagents to start our syntheses. The just-in-time (JIT) procurement system permits us to have most chemicals at our doorstep within 18 to 24 hours. Even so, detailed planning and logistics go into making sure all reagents and solvents and gases and glassware are ready for a day's lab work. Solvents need to be pre-distilled before use, since small impurities can promote or catalyze undesired side reactions. Intermediate molecules need to be pre-made and properly stored in a freezer away from light and oxygen to prevent their decomposition while the other segments of the synthesis are being done. A rich chemical literature provides guidance on reaction types and conditions that are usable on similar molecular constructs, although modifications are almost always needed since the substrates in a new synthesis are different. None of this would have been possible on the prebiotic Earth. As a further difficulty, reagent addition order is critical. A needs to be added before B, and then C, and each at its own specific temperature to effect a proper reaction and coupling yield. The parameters of temperature, pressure, solvent, light, pH, oxygen, moisture, have to be carefully controlled. Unless one can devise sophisticated promoters or catalysts that are stable in air and moisture and can work at common atmospheric conditions, precise control must be maintained. But making such ambient stable promoters or catalysts is more complex than just varying the temperature for the specific reagent, or just putting the reaction in a carefully maintained atmospheric control box, dry box, equipped with oxygen and moisture sensors, all maintained under a positive pressure of inert nitrogen gas. It's not cheap. Once the desired product is synthesized, it can take much longer to properly characterize the product than it did to make it. We use many tools, costing millions of dollars, to facilitate rapid molecular structure identification. As teachers is the great Scotch, water is the great solvent. But organic synthesis is very hard to do in water. Highly oxygenated organic compounds are needed. The synthetic chemist must project the oxygenated groups out toward the water domain, and project the non-oxygenated groups in toward each other, thus generating a hydrophobic domain. It is very hard to do. By doing our nanocar organic synthesis in organic solvents rather than in water, we markedly lessen the difficulty. It is a luxury that nature did not, and does not, enjoy. Starting from scratch, she would have had to design and redesign her structures, discarding the inevitable false starts and dead ends as they occurred. Any prebiotic system is destined, at least some of the time, to crash and burn. How would nature know where to stop, or how to start over, with no goal in mind? But whatever else she may have been doing in the prebiotic era, nature was not consulting the modern chemical literature. Life lessons for the prebiotic chemist. To illustrate that the problems I describe above are not limited to exotic structures such as nanocars, let's examine the synthesis of foundational organic molecules, carbohydrates. Carbohydrates are the backbones of nucleotides, which in turn make up DNA and RNA. Carbohydrates also serve as recognition sites for cells to communicate with each other, and as food sources for living systems. The difficulties involved in carrying out carbohydrate synthesis in a prebiotic environment parallel those found in making nanovehicles. Consider the pentose sugars, carbohydrates with 5 carbon atoms, Fig. 4.6. These sugars have 3 stereogenic centers, so there are 8 possible isomers. Some substructures are enantiomers, mirror images, others diastereomers, not mirror images, all are chiral. When we carry out chemical reactions, we design the reactions to minimize diastereomeric mixtures that can be nearly impossible to separate. We try our best to avoid the undesired diastereomers because their separation is too time-consuming and expensive. They waste a huge amount of starting material, they generate unwanted products. Enantiomeric separations are all the more difficult. Nature has apparently chosen a far harder route, using predominantly one enantiomer, homochiral, D-ribose, in a system with multiple stereogenic centers. So how difficult would the synthesis of D-ribose be under prebiotic conditions? Albert Eschenmoser is a great synthetic chemist. He has spent years suggesting prebiotic routes to the 5-carbon pentoses. Direct synthesis of ribose, he discovered, was not successful when starting with glycoaldehyde, using an old-fashioned Formose reaction, in which a base is catalyzed with formaldehyde in the presence of a divalent cation such as calcium. To synthesize ribose, Eschenmoser had to make phosphorylated glycolaldehyde phosphate, figure 4.7. Since glycolaldehyde is the dimeric form of formaldehyde, he first had to make the dimer of formaldehyde. Only then could there be further aldol chemistry in the Formose reaction. A good organic chemist can design conditions that will isolate the product, purify it, and then proceed. A very good organic chemist, this is what Eschenmoser did. Can phosphorylation then occur? Yes, but only given high concentration of a phosphorylating agent. It does not happen in the presence of the strong base required for the formation of the glycoaldehyde itself. The glycoaldehyde needs a strong base to pump up the reaction, and then, to stop it dead, it needs to be separated from that strong base. While in a near-neutral aqueous solution, and in the presence of magnesium ions, Eschenmoser phosphorylated glycoaldehyde with 4 molar equivalents of amidotriphosphate. Once phosphorylation was complete, and the glycolaldehyde phosphate isolated and purified, Eschenmoser then exposed glycolaldehyde phosphate to the very basic 2N-sodium hydroxide at a concentration of 0.08 moles per liter. In other words, the formaldehyde started life in a strong base, its product was isolated and freed from the strong base, and exposed to a neutral aqueous solution of amidotriphosphate, which the researchers made separately. That product, glycolaldehyde phosphate, was then isolated and conveniently re-exposed to a strong base. Even with this masterful design, the result was mostly undesired racemic hexose triphosphates. Using a stroke of superb synthetic insight, Eschenmoser placed the glycolaldehyde phosphate in the strong base, and then added 0.04 moles per liter of formaldehyde to obtain a 40 to 50% yield of mostly racemic pentose diphosphates, the mixture shown in figure 4.7, but with 2 phosphates at C2 and C4 of each of the structures. In that 40 to 50% yield mixture, there were then 8 possible isomers: 4 diastereomers, the desired racemic ribose, 2,4-diphosphate, tilde 15% of the total from the reaction, racemic arabinose, 2,4-diphosphate, racemic glycose, 2,4-diphosphate, and racemic xylose, 2,4-diphosphate, where racemic signifies a 1:1 mixture of the 2 enantiomers, along with 11 other identified carbohydrate species, all bearing their enantiomer partners. That meant 22 other identified species from the 40 to 50%, with the remaining 50 to 60% being unidentified non-volatile compounds such as higher oligomers and polymers. Even Eschenmoser did not attempt to separate out the desired, albeit still racemic, ribose, 2,4-diphosphate. And for very good reason, it would have been nearly impossible. Biologists can easily imagine nature selecting the correct isomer because they work in a world that enjoys the specificity of biological systems. Not so synthetic chemists, who are bound to prebiotic molecules. Selected by what? No enzymes were yet available. The data more readily suggests that no prebiotic process is likely to yield the requisite carbohydrates. The most masterful of synthetic chemists could produce only gross mixtures. Also, time works against life. Over a mere 23 weeks, the desired diastereomer, the racemic ribose, 2,4-diphosphates, was reduced from a 17% yield to a 7% yield. After a year, there would be very little left. In the laboratory, as anywhere else, it is essential to stop a reaction before the desired product degrades. Of all the isomers, the one desired was not the most stable, even given the cleverly designed experiment that started with glycolaldehyde, and that was further intelligently and conveniently phosphorylated to the glycolaldehyde phosphate. Racemic ribose, 2,4-diphosphates degraded under the very controlled conditions under which they formed. Eschenmoser writes that the total amount of the 4 diastereomers is largely unchanged, but his own data shows that the combined yield of the 4 diastereomers dropped from 34% to 30% over the course of the additional 22 weeks, a relative 12% loss of the pentoses over 22 weeks. After a few years, a brief moment in time in prebiotic terms, there would be few if any of the pentoses left, let alone the desired ribose, 2,4-diphosphate. Where does the material go? It likely degrades to extended oligomers and polymers, a process common in organic reactions, and especially common in extended aldol-based reactions. Wish fulfillment. From a paper on prebiotic chemistry. Moreover, there was the well-known, but still no less remarkable, fact that in cellular biochemical processes, monosaccharides apparently never operate in the free state, but always in phosphorylated form. It is a short step from such considerations to the notion of a primordial scenario in which, again, phosphorylated and not simply neutral forms of carbohydrates would have been operative, emphasis added. In a self-organization process in a primordial environment, it may have been of primary importance for carbohydrate molecules to escape chemical chaos, finding themselves instead at concentrations suitable for chemical reactions, and in reaction spaces that would facilitate efficient chemical transformation. With respect to both requirements, phosphorylated sugar molecules would, through their electrical charges, have offered advantages over neutral, water-soluble carbohydrates in environments containing mineral surfaces or minerals with expandable layer structures. That short step is not short at all. Biochemical routes are far downstream and occur in far more complex scenarios. In the laboratory, phosphorylation requires precise control of phosphorylating agents. These hopeful but unlikely suggestions pain the synthetic chemist under any circumstance, but for some remarkable reason, they are tolerated in prebiotic chemistry. Despite claims to the contrary, research on mineral surfaces has done little to solve the problem of overall yields, or that of diastereo and enantioselectivities. Reagent addition order is critical. An abiogenetic pathway would require several lines of intermediates forming in proximity, and then coming together in the proper order at the precise moment and location needed for synthesis. We could modify many parameters during synthesis: temperature, pressure, solvent type, light, pH, oxygen, moisture. No such controls figure in a prebiotic environment. Characterization is critical. Without it, impurities accumulate. What prebiotic characterization might mean is anyone's guess. Given poor prebiotic reaction yields, it is impossible to envision a process in which the starting materials generate all of the desired products. In synthesizing nanocars, we had to go back over and over again to generate molecular intermediates, a process known familiarly as bringing up material from the rear. How would prebiotic chemistry bring up its own rear over and over again? It has kept no laboratory notebook to record the previous paths. In our synthesis, we spent a great deal of time on separation and optimization. If byproducts are permitted to accumulate, they often will consume the new step's reagents and alter the course of the reaction. This problem would plague abiogenesis, too. It stands to reason. There must have been a chemical means, once upon a time, to generate an information-bearing molecule such as DNA or RNA. Since the 1960s, a number of biologists have suggested that the polymer is RNA rather than DNA. Such is the RNA world hypothesis. And chemically activated ribonucleotides can polymerize to form RNA. So far so good. But RNA is far less stable than DNA, and whatever the polymerization, it yields generic RNA, a molecule lacking sequence specificity. Had RNA researchers succeeded in producing a volume of random sentences, example, subtends flac lachrymose esurient, none of them would have imagined that they had succeeded in composing King Lear. The coupling of a ribose with a nucleotide is the first step, and even those engrossed in prebiotic research have difficulty envisioning that process, especially for purines and pyrimidines. John Sutherland and his co-workers have proposed that pyrimidine ribonucleotides can form short sequences using arabinose aminooxazoline and anhydronucleoside intermediates, all from simple compounds such as cyanamide, cyanoacetylene, glycolaldehyde, glyceraldehyde, and inorganic phosphate. The use of inorganic phosphate changes the experiment's basic conditions to a pH-buffered solution, thereby slowing decomposition pathways. But the work itself shows the intricacies required to generate the desired reactions. The conditions they used were cleverly selected. Although the issue of temporally separated supplies of glycolaldehyde, just-in-time, JIT, Scenario 1, and glyceraldehyde, JIT Scenario 2, remains a problem, a number of situations could have arisen, prebiotically, what and where. That would result in the conditions of heating, careful control of step 1 at 60 degrees Celsius, and progressive dehydration, careful control of step 2 by lyophilization, which is water removal by ice sublimation under reduced pressure of less than 0.001 atmospheres, followed by cooling, careful control of step 3 from 60 degrees Celsius to 23 degrees Celsius, rehydration, careful control of step 4 with precise adjustments of pH, and ultraviolet irradiation, careful control of step 5 with a selected 254 nanometers light. There were also multiple purification steps, careful control of step 6, and ion exchanges using commercial resins, careful control of step 7. All this for the synthesis of just one set of a mixture of adducts, and in racemic form. It remains clear that the controlled conditions required to generate even a mixed set of select structures is painfully improbable. Routes to each one of the requisite carbohydrates, lipids, nucleic acids, and proteins, polymers of amino acids, have been proposed in prebiotic studies. The attempted syntheses almost always create mixtures beset with the same difficulties. From the data, the synthetic chemist can easily deduce that under prebiotic conditions the reaction in question is not likely to yield anything useful. With each added step, difficulties are compounded by improbabilities so overwhelming that no other field of science would depend upon such levels of faith. Abiogenesis research would never be accepted in any other area of chemistry. Extrapolation on steroids. But this is not the end. Making carbohydrates, lipids, amino acids, and nucleic acids still has not built a cell. Sutherland and co-workers pointed out in 2015 that a minimal cell can be thought of as comprising informational, compartment-forming and metabolic subsystems. They also acknowledged that, to date, prebiotic chemistry has made ambitious extrapolations to imagine the abiotic assembly of such an overall, cellular, system or subsystem, however, places great demands on hypothetical prebiotic chemistry. Yet this revealing comment by Sutherland and his co-workers is coupled with their disclosure of a new experimental finding showing that precursors of ribonucleotides, amino acids and lipids can all be derived by the reductive homologation of hydrogen cyanide cyanide and some of its derivatives. The key reaction steps are driven by ultraviolet light, use hydrogen sulfide as the reductant and can be accelerated by Cu(I), Cu(2) photoredox cycling. They assert boldly that all the cellular subsystems could have arisen simultaneously through common chemistry. This has now raised the level of suppositions from mere molecule types to complex subsystems where molecules are working in concert toward a common functional goal. But compositions of a few molecule types, or even all of them, do not constitute a cellular subsystem. It is essential to emphasize that the authors only prepared precursors to the ribonucleotides, amino acids, and lipids, not the actual molecules, so the gross extrapolation is all the more disconcerting. When reading the protocols for the suggested prebiotic-like precursors, one is struck by the high-level sophistication, expert synthetic prowess, and remarkable ingenuity of the researchers. Some reactions were run at room temperature, some at 60 degrees Celsius, others at 100 degrees Celsius and then washed with ice-cold water. Often the molecules prepared by these supposed prebiotic routes were not used, but had to be made more cleanly and in larger scale using purely synthetic methods and organic solvents, such as Lawson's reagent and tetrahydrofuran, respectively, to simplify the handling procedures. JIT and precise order of addition protocols were used over and over again. One sees precise pH adjustments through the syntheses, use of ion exchange resins, and separations from the reaction mixtures because proceeding without separations would have destroyed the carefully prepared products. The preparation of cyanoacetylene on Cu(I) was suggested as a way to prepare it conveniently and store it for use when needed. CuCl was mixed with KCl to generate the Newland catalyst, KCuCl2, at 70 degrees Celsius. Then a separately generated source of acetylene gas was prepared from CaC2 and water. This gas was bubbled through the Newland catalyst to prepare acrylonitrile, an unstable molecule that needs proper isolation and storage to inhibit its polymerization, which was then treated with KCN for 1 hour, then 5 equivalents of NH3 as a 13-molar NH3, NH4+ solution adjusted to pH 9.2 with NaOH to generate the desired aminopropionitrile. All of the reactions were executed in separate clean vessels and properly isolated prior to proceeding to the next reaction. This is just a sampling of preparations that are difficult even for the skilled synthetic chemist to execute. The routes afford very simple precursors to just a few of the many molecules within the building block classes of molecules, carbohydrates, lipids, amino acids, and nucleic acids. Finally, all the precursors were racemic if they even bore any possible stereoisomerism. Dream on. The world's best synthetic chemists, biochemists, and evolutionary biologists have combined forces to form a dream team in two quite distinct senses of the word. Money is no object. They have at their disposal the most advanced analytical facilities, the complete scientific literature, synthetic and natural coupling agents, and all the reagents their hearts might desire. Carbohydrates, lipids, amino acids, and nucleic acids are stored in their laboratories in a state of 100% enantiomeric purity. Would the dream team, please, assemble a living system? Take your time, folks, take a few billion years. Nothing? Well. Let us assume that all the building blocks of life, and not just their precursors, have been made to a high degrees of purity, including homochirality where applicable, the carbohydrates, the amino acids, the nucleic acids, and the lipids. They are stored in cool caves, away from sunlight, and away from oxygen. These molecules are indifferent to environmental degradation. And let us further assume that they are all stored in one comfortable corner of the Earth, not separated by thousands of kilometers or on different planets. And that they all exist not just in the same square kilometer, but in neighboring pools where they can conveniently and somehow selectively mix with each other as needed. Now what? How does the dream team assemble them without enzymes? Very well. Give the dream team polymerized forms, polypeptides, all the enzymes they desire, the polysaccharides, DNA and RNA in any sequence, cleanly assembled. Ready now? Apparently not. We teach our students that, when a mechanism does not support their observations, the mechanism must either be revised to support the facts or entirely discounted. They are not required to provide an alternative. Those who think scientists understand how prebiotic chemical mechanisms produced the first life are wholly misinformed. Nobody understands how this happened. Maybe one day we will. But that day is far from today. It would be far more helpful and hopeful to expose students to the massive gaps in our understanding. Then they may find a firmer, and possibly a radically different scientific theory. 5. Digital Evolution, Predictions of Design. Winston Ewart. Summary. Computer simulations of evolution are often invoked in defense of the abilities of Darwinian evolution. A number of well-known simulations are discussed, showing how they follow the predictions of intelligent design in requiring teleological fine-tuning in order to work. This and other predictions of intelligent design have been confirmed by simulations, whereas Darwinian evolution offers no predictions about computer simulation and is thus unfalsifiable. Introduction. In Chapter 2, by Stephen Meyer, we saw that the neo-Darwinian mechanism of mutation and natural selection does not provide an adequate explanation for the origin of the genetic information necessary for building new forms of life. Lacking biological evidence for the creative power of this mechanism, some proponents of neo-Darwinian theory have sought to simulate the creative power of the mutation selection mechanism using computers. Indeed, computer simulations are a growing part of the debate over Darwinian evolution. Proponents of Darwinian evolution have developed an array of computer simulations, arguing that they demonstrate evolution in action, confirming their theory. Critics of evolutionary theory have developed their own simulations, arguing the exact opposite, that evolution does not work in practice. Before looking at these simulations or arguments, we need to take a step back and look at how we decide whether or not a scientific theory is true. Scientific theories are difficult, usually impossible, to logically prove because they make universal claims. For example, a theory might claim that all swans are white or that there are no perpetual motion machines. These are called universal claims because they are about all swans or all machines. It is relatively easy to check whether a particular swan is white or a particular machine exhibits perpetual motion. However, no matter how many swans or machines are investigated, this will never prove that all swans are white or that no machines exhibit perpetual motion. We simply cannot prove a universal law by looking at examples, no matter how numerous. Falsification is one possible solution to this problem. If a theory is false, then there should exist counterexamples. When a counterexample to a theory is found, we say that the theory is falsified. At that point, we know that the theory is false. On the other hand, if after searching long and hard for a counterexample, we are unable to find one, we can conclude that the theory is probably true. We do not know for certain that the theory is true, it remains unproven, but it is increasingly unlikely that a counterexample exists and thus increasingly likely that the theory is correct. A falsifiable theory takes on a certain risk by offering the possibility of being refuted by a counterexample. Not all propositions do this, however. For example, one could claim that the universe sprang into existence last Thursday. All memories and artifacts appearing to be from before that time were simply part of the world when it sprang into existence. For example, if you found rings in a tree, you might think this proved the age of the tree. However, one could explain this away by claiming that the tree sprang into existence last Thursday already containing the tree rings. There is no counterexample that can be presented to demonstrate that this claim is incorrect. Thus, we say that the claim is not falsifiable. We would not expect to find a counterexample, even if the theory was false. Since no possible counterexample can exist, even in principle, the theory takes on no risk of being falsified, and thus the lack of counterexamples does not provide evidence for the theory. Such reasoning can never logically prove that a theory is correct. It is always possible that a counterexample exists but has not yet been found. There may yet be a perpetual motion machine that nobody has figured out how to build. Nevertheless, given the large amount of effort spent attempting to build perpetual motion machines, all ending in failure, we can be reasonably confident that a perpetual motion machine cannot exist. At the very least, we can provisionally accept the theory until a counterexample is produced. There are other reasons to accept or reject a theory. One might reject a theory as too complicated, invoking Occam's razor. Or a theory might be proved by a mathematical theorem, thus being accepted without requiring any empirical evidence. For example, under appropriate assumptions, perpetual motion machines are proven impossible by Noether's theorem. Nevertheless, for this chapter, we will focus only on falsification. How do computer simulations fit into the process of falsifying theories? A computer simulation can falsify a theory, but only if that theory makes a claim or prediction about the simulation. If I were to claim that Darwinian evolution would not work even inside a computer simulation, then a single example of successfully simulated Darwinian evolution would prove my claim incorrect. But most theories, including that of intelligent design, are about the real world, not computer simulations. They do not make explicit claims about what will happen in any simulation. As long as a theory makes no claims about computer simulations, no computer simulation can provide a counterexample. However, in many cases, a theory implies predictions about computer simulations. The theory may not offer an explicit prediction about computer simulations, but an implied prediction may be a necessary consequence of the theory being true. In that case, we can use the simulation to falsify the theory by providing a counterexample to that implied prediction. Care must be taken, however, to consider only predictions actually implied by the theory, and not merely predictions attributed to a theory, especially by its critics. In particular, critics of intelligent design have attributed predictions about computer simulations to intelligent design that intelligent design proponents have not made, would not make, and that are not implied by intelligent design theories. These are counterfeit counterexamples, and they do not falsify the relevant theories. In this chapter, we will look at actual predictions implied by intelligent design with respect to computer simulations. We will show how various alleged counterexamples actually fit the predictions of intelligent design. Thus, for all of the attempts at falsifying intelligent design through computer simulations, the intelligent design predictions have survived these tests, thereby providing evidence for intelligent design. 2. What are computer simulations of evolution? What does it actually mean to simulate Darwinian evolution? In other fields, scientific simulations typically seek to closely imitate and thus predict real-world phenomena. For example, modern weather prediction is based on detailed computer simulations of changing temperature, wind speed, pressure, and so on. Likewise, new engineering designs are often tested in computer simulations to discover flaws before construction. Because these simulations adhere closely to reality, they can offer predictions about it, and we can verify the accuracy of the simulations by comparing them to reality. Evolution simulations are not in the same category. A weather simulation predicts whether or not it will rain tomorrow, but no evolution simulation can predict whether or not a rabbit will evolve a faster hop, a lion, sharper teeth, or an ape, language skills. Evolution simulations rarely, if ever, make any predictions about real-world biological evolution. Instead, computer simulations of evolution reduce Darwinian evolution to its essence, and then observe the resulting process. This allows us to learn about Darwinian evolution divorced from its biological context. But what is this essence of Darwinian evolution? According to philosopher Daniel Dennett, evolution will occur whenever and wherever three conditions are met: replication, variation, mutation, and differential fitness, competition. Theistic evolutionist Kenneth Miller says, what's needed to drive this increase? Just three things: selection, replication, and mutation. It's no coincidence that the same three things are required for evolution, since what we are observing is nothing less than evolution on a small, observable scale. Richard Lewontin wrote, the generality of the principles of natural selection means that any entities in nature that have variation, reproduction, and heritability may evolve. Replication or heritability means that offspring resemble their parents more than other members of the population. Children look more like their parents than their parents' friends. This is true throughout the biological world, from humans to dogs to fishes to birds, and even to viruses. Variation or mutation means that, while offspring are similar to their parents, they are not identical. Instead, there are random variations or changes. Selection or differential fitness refers to the propensity of some members of a population to reproduce more than others. As an example, the faster gazelle does not get eaten, and thus has more offspring. At least according to the Darwinists quoted above, these three factors are the essence of Darwinian evolution. Any evolutionary simulation, seeking to incorporate the essence of Darwinian evolution, works by including these three processes. To better understand the concept, here is an exercise that you can do at home. For this, you will need 10 coins and a die. Take 5 coins, and place them face down on a table. This represents the first living cell in your very own virtual world. The sequence of heads and tails coins is its DNA. First, we need to include replication. Our cell or row of coins must split into 2 rows of coins, both of which resemble the original row. To model this, take the remaining coins and create a second row of 5 coins, exactly like the first row. Since your first row of coins are all face down, the new row will also be all face down. Second, we need mutation. The copies should not be exactly like the original row. They must vary in a random fashion. To model this, roll a die twice, once for each row. Flip the coin corresponding to the number that you rolled. For example, if you roll a 3, flip the third coin in the row. If you roll a 5, flip the fifth coin. If you roll a 6, you will notice that there is no sixth coin, and you should not flip any coin. Third, we need selection. Some rows of coins should survive better than others. You should currently have 2 rows of 5 coins, and you want to determine which row is fitter. For the sake of this example, we will arbitrarily decide that more heads is fitter. In the case of a tie, break the tie randomly, it does not matter. The fitter cell, row of coins, survives, but the less fit cell dies. Take it, i.e., the less fit row of 5 coins, off the table. At this point, you are back to having only one cell, one row of coins. Repeat the process. The cell row splits, resulting in 2 identical cells rows of coins. To do this, you'll need to bring the discarded coins back to the table, this time matching the row left on the table from the first round. Roll the die twice, flipping the corresponding coin for the numbers rolled in each row. Keep the fittest row, the one with the most heads, removing the other one. Repeat until bored or until the row has evolved into all heads. I did this experiment, and at the beginning of each iteration I recorded the sequence of heads and tails in my row. For each heads, I wrote H, and for each tails, I wrote T. I ended up with the following sequence. TTTTT. TTHTT. THHTT. THHTT. THHHT. HHHHT. HHHHH. Evolution started with a sequence of coins that was all tails up, however, it rapidly evolved into one where the sequence was all heads up. The process exhibited the 3 essential conditions of Darwinian evolution. Each child was similar to the parent, as required for replication. However, they were not exact copies, as required by mutation. Finally, the rows with more heads in them had more children, thus fulfilling selection. This is a very simple model. The other models that will be discussed in this chapter, as well as others not discussed, are more sophisticated, but the essence comes down to the same idea. The simulations all apply replication, mutation, and selection to evolve artificial organisms. The details in each model will vary greatly, but this is the core of what makes these models work. 3. Design requires intelligence. Intelligent design makes the falsifiable claim that design, or the appearance of design, derives only from intelligence. When we observe complex processes, digitally encoded information, or finely tuned machines, we observe the appearance of design, and according to the theory of intelligent design, we have good reason to infer intelligence. Stephen Meyer argues, if we trace information back to its source, we always come to a mind, not a material process. Meyer's argument is a falsifiability argument. If anyone were to present a counterexample, design without intelligence, then intelligent design's claim would be falsified and would have to be abandoned. However, continued absence of a counterexample provides evidence for intelligent design. According to Darwinist arguments, however, there are counterexamples to this claim: computer simulations. Computer simulations have evolved everything from English phrases to antennae to computer programs. If we were to find any of these in nature, intelligent design proponents would immediately infer intelligence as the best explanation. These are instances of the appearance of design. Thus, according to the argument, design can be produced without intelligence, and therefore intelligent design is falsified. However, recall that intelligent design's claim is about the real world, not about computer simulations. As such, it may seem that the outcome of a computer simulation is irrelevant to intelligent design's claims. However, a computer simulation is, in essence, a machine located inside the real world. A simulation that evolves an English phrase is a machine that produces that English phrase. A simulation that evolves a computer program is a machine that produces that computer program. If we have a simulation that produces an artifact appearing to be designed, that means that there exists a machine that can produce design. That machine exists in the real world, and thus the claims of intelligent design apply to it. But this machine would seem to be exactly what intelligent design claims cannot exist, a device that can produce design without intelligence. This is incorrect, however, because intelligent design does not argue that machines cannot produce design, rather, it argues that only intelligently designed machines can produce design. As an example, consider the machine that produced this book. This book appears designed, it contains much, hopefully, meaningful text. But it was produced by a printing press, not by a human writing out the words by hand. There was no intelligence at all involved in the physical production of this book. However, the printing press itself was designed, and the words derive not from the machine but from this book's authors. Intelligence was required, not in the physical construction of the book, but in the configuration and inputs of the machine that produced this book. Every computer simulation is intelligently designed. These simulations do not simply arise on unused computer hardware. In every case, programmers deliberately design them. Despite being designed in imitation of nature, a computer simulation could no more arise spontaneously than could a robotic car factory. Thus the intelligent design claim is verified. It took intelligence to produce machines that could produce design. On the other hand, while the simulations are designed, they are imitations of nature. They have taken what is in nature and translated it into a digital realm. It would be nonsensical to attribute the insights of Sun Tzu to a 20th century translator. Likewise, it is argued to be nonsensical to attribute the success of a computer simulation to its programmer when the programmer was merely translating the natural world into a digital realm. Rather, the success must be due to what was translated. However, that is not how translation actually works. While Sun Tzu's insights are his own, much of the quality of an English translation is due to the translator rather than Sun Tzu. Sun Tzu could have written brilliant Chinese that was mangled beyond understanding by a translator, or he could have written horrific Chinese that was translated into beautifully readable English. This is most obvious in the case of poetry, which commonly uses devices such as rhyme or rhythm that do not easily translate, and can be present in the translation only by the talent of the translator. When the programmer translated nature into a computer program, is the resulting success due to the source or to the translator? Does evolution work in a simulation because it works in nature, the same way insights are in a translation because they were first in the original text? Or does evolution work in a simulation because of the talent of the programmer, like a talented poet producing beautiful poetry while translating the work of a talentless author in another language? In order to differentiate these possibilities, we look for examples of teleological fine-tuning in a simulation. Teleological fine-tuning refers to cases where a system is designed, configured, or built in a particular way for the purpose of producing a desired outcome. Teleological means that something was done for a particular purpose, in order to achieve some end. Fine-tuning means that the system was configured in such a way that it has some particular property, as opposed to alternative configurations that would not have had that property. Consider the case of a bucket of water perched on a door by a prankster, such that when the door opens, the bucket falls and pours water on the person who walked through. The system is fine-tuned, because for most possible locations of the bucket, it would not soak the person walking through the door. Furthermore, it is teleological because the prankster placed the bucket in that position specifically so that it would fall and soak the next person who walked through the door. It is thus an example of teleological fine-tuning. If a simulation contains teleological fine-tuning, the intelligent design claims stand. It took a goal-directed intelligent agent to produce the teleological fine-tuning in the simulation. Such simulations are not translations of nature but are cases where the translator used his or her own intelligence to produce a working simulation. On the other hand, if a simulation contains no examples of teleological fine-tuning and yet produces design, then the claims of intelligent design are falsified. This leaves us with the question, are there examples of simulations that avoid teleology but still successfully demonstrate the evolution of the appearance of design? To answer that question, we need to look at examples. We will investigate a number of simulations, showing their teleological fine-tuning. 4 examples. A. Dawkins's Weasel. As a first example, consider a simulation presented by Richard Dawkins in his book The Blind Watchmaker. Dawkins does not give his simulation a name, but it is often known as Dawkins's Weasel. It is not a particularly good simulation, even Dawkins quickly points out its shortcomings. However, it is a simple model and readily understood, thus providing a good starting point for this discussion. The goal of the Weasel simulation is to evolve the phrase, "Methinks it is like a weasel." The phrase itself is a quote from Shakespeare's Hamlet, where the characters are discussing which animal a cloud looks like. The simulation begins by selecting 28 random letters and spaces. In the run discussed by Dawkins, he obtained WDMLT-DTG-QMCOP. This is pretty much what one would expect by selecting a sequence of random characters. However, Dawkins's Weasel does not stop there. It generates many slightly mutated copies of the original phrase. Dawkins does not say how many copies are made or how exactly they are mutated. Nevertheless, making 100 copies of the string, each one with a single character randomly replaced, produces results that are commensurate with the examples presented in Dawkins's book, and while we do not know whether this was the original algorithm, for the sake of the argument we will assume that it was. At this point, the simulation has 100, or so we assume, slightly modified copies of the original string of characters. The simulation picks the copy that is closest to the target. The closer to the target, the fitter the copy is deemed to be. How do we measure how similar a string is to the target? There are, in fact, a number of different ways to answer this question. Dawkins used what is called the Hamming distance. To compute the Hamming distance, we line up the two strings of interest, identifying where the two strings are different. Methinks it is like a weasel WDLTMNLT-DTG-QMCOP. XXXXX20XXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXXX the two upper strings are different. The 3 blank spaces are where the random string and the target string happen, by chance, to agree. In this case, there are 25 XS, giving the random string a Hamming distance of 25 from the target. Compare this to another string later in Dawkins's run. Methinks it is like a weasel meldinals it Isaac Z Wexel 20xxxxxx. This case has only 8 points where the string is different from the target. Thus it has a Hamming distance of 8. Since 8 is less than 25, this string is much closer to the target than was the original string. Having used the Hamming distance to establish which of the 100 copies is closest to the target, the simulation throws out all copies except the closest. In the case of Dawkins's example run, while the original random string was WDMLT-DTG P, the best of its 100 copies was WDMLT-DTG's ResRemco P. Detecting the difference between these two strings is difficult for the human eye, but the second phrase has replaced a DTJBK with a DTJBS, which is closer to the target because the S is in the correct place to form the word IS. The process is then repeated, with this newly adopted copy as the original for 100 new copies. After 43 iterations of this copy-mutate select process, Dawkins found it converged onto his target. Methinks it is like a weasel. Dawkins reported running the algorithm a second time, this time taking 64 iterations. In either case, the simulation readily produced the target phrase. But does this simulation exhibit teleological fine-tuning? To see, we need to go back through the process used by the simulation, carefully examining each step for instances of teleological fine-tuning. The first action of the simulation is to pick 28, the length of the phrase, methinks it is like a weasel, including both spaces and letters, characters at random. What could be less teleological than a completely random starting point? Indeed. However, while the selection of the letters was random, they were chosen from a non-random set of possibilities, the 26 letters of the English alphabet and a space. Why include spaces but not digits, punctuation, or other symbols? Of course, the simulation had to pick some collection of possible symbols, but the selection of this particular set of symbols was teleological. The set of possible characters was chosen specifically because it was the set of characters used in the target. Dawkins could have included digits, symbols, punctuation, and even letters from other alphabets. Instead, he used only the types of characters he needed in order to reach the target. Additionally, 28 characters were chosen. Why 28? Obviously, 28 was chosen because that is the number of characters in the target. If he had chosen a different number of characters, then the simulation would not have found the target. Dawkins started with 28 characters because he knew that was what he needed in order to reach the target. We do not know how many copies of the string are made in Dawkins's simulation, but in order to replicate Dawkins's results the number of copies has to be large, around 100. Why has Dawkins chosen to make a large number of copies rather than a small number? More copies mean more chances to find beneficial random mutations. He has chosen a number that allows his simulation to quickly reach the target. Dawkins reported 2 runs of his simulation, one taking 43 generations and the other 64. However, my own simulation, which makes only 10 copies, took 723,232 generations in one attempt and 461,300 in another. A seemingly minor choice in the design of the algorithm made Dawkins's algorithm 10,000 times faster. Mutations appear to take the form of randomly replacing a letter in the string with another. Why simple replacement and not insertion or deletion? Dawkins did not include those because he knew they were not needed in order to reach the target. Since he began with a string of the correct size, there was no need to have mutations that could change the size. In each iteration, the simulation selects the copy closest to the target as the fittest. Why is that the fittest? Why is WDMLT-DTG's res-rem copy fitter than WDMLT-DTG querm copy? Certainly, there is no more sense or meaning in the one than the other. Neither is functionally better than in the other, in any way. The only basis for preferring the one to the other is teleological. The one is closer to a predefined goal than the other. Dawkins does not dispute this point. He readily acknowledges that imitating selection by measuring distance to a target is not a proper model of evolution. As he says, life isn't like that. Evolution has no long-term goal. He freely acknowledges a teleology to this model that is inappropriate to a model of Darwinian evolution. We have looked, in perhaps mind-numbing detail, at the various aspects of fine-tuning that went into constructing Dawkins's weasel. This was somewhat unfair, since Dawkins described this simulation as misleading in important ways. Dawkins intended the algorithm only to make his point about the distinction between single-step and cumulative selection, not to make a case for Darwinian theory. Nevertheless, it provides a useful example as an introduction to the exploration of teleology in evolutionary simulation. We have seen that at almost every part of this simulation, teleological fine-tuning was present to guide it to its target. BEV. Moving on from Dawkins, consider Thomas Schneider and his program EV. Whereas Dawkins was careful to note some limitations of his weasel simulation, Schneider argues that his simulation utterly destroys opposing arguments. The EV model shows explicitly how this information gain comes about from mutation and selection, without any other external influence, thereby completely answering the creationists. Understanding EV will require some background. Imagine that, after having read this book, you are skimming through it again in hopes of finding a particular section that you found interesting. The section in question was about dolphins, and thus you skim through the book looking at each word only long enough to ascertain whether or not it might be the word dolphin. Once you have found the section, then you actually read it carefully. This is somewhat similar to the process that a cell uses when reading DNA. While this book is a sequence of English letters, DNA is a sequence of nucleotides. The nucleotides are typically represented as the letters A, C, G, and T. The cellular machinery skims through the DNA looking for particular patterns of A, C, G, and T's. For example, it might look for the pattern to tart. Actually, it looks for tart or a number of similar sequences. Molecules attach to the DNA at the point of these patterns, which are called binding sites, causing some desired effect. For example, it might increase or decrease the amount of a particular protein being produced. EV models this process. One part of EV's genome defines the patterns that are recognized as binding sites. This is done using a structure known as a perceptron. The details of how a perceptron works are not important here, simply that it defines a set of patterns that will be considered binding sites. The other part contains 16 positions where a binding site should exist. The binding sites will actually exist only if the pattern of nucleotides at that position fits one of the patterns accepted by the perceptron. The problem is then to identify both a perceptron and a sequence so that the correct binding sites, and only the correct binding sites, are recognized as binding sites. EV evolves a solution to this problem. For each organism, it counts the number of mistakes. Mistakes are binding sites that are missing or are in an incorrect position. The half of the population with the fewest mistakes is kept, but the half with the most mistakes is killed. The eliminated half of the population is replaced with a mutated copy of the surviving half of the population. This mutate-select-replicate process quickly results in a solution to EV's problem. Is this a teleological model? EV models the evolution of a real biological structure, a binding site, in DNA, which makes it closer to biology than any of the other models considered in this chapter. However, at a few key points, EV deviates from the biology and employs intelligently designed systems to help it reach its target. The most important point of teleology in this model is the way in which the simulation decides which organisms are fitter. An organism is fitter when the binding sites in the organism are closer to the correct binding sites. The correct binding sites are a target, and EV is constantly measuring how far away any particular organism is from that target. But this is a massive departure from biology. Natural selection evaluates how well an organism works in its present environment. It does not measure the distance between an organism and some ideal form. The ability to measure the distance to a target is very powerful. Imagine that you have a dog that has escaped. Fortunately, the dog has a transmitter on his collar, and you have a receiver. The receiver tells you how far away the dog is, but not the direction. Nevertheless, there will be no problem in locating your dog. You simply have to walk in the direction that causes the number on the receiver to go down. What will happen without this transmitter? Finding your dog is going to be much harder. There may be other pieces of information you can use, such as where your dog has gone before, where other dogs are, where you have caught glimpses of your dog, and so on. But all of those are going to be much less useful, much less powerful, than being able to measure the distance. While EV models biology in the object of evolution, binding sites, it does not follow biology in its model of natural selection. Instead, it follows the unavoidably teleological method of measuring the distance to a target. This is critical. Evolution succeeds or fails depending on how well natural selection performs in guiding the evolutionary process. Replacing selection with a teleological process, as done by EV, eliminates any claims it might have had to being based on biology rather than being teleologically driven. See Steiner trees. Another example is that of Dave Thomas, who wrote a simulation that evolved solutions to the Steiner tree problem. The problem is essentially that of a collection of cities wishing to build a road network between them. The road must be built with the least amount of cost, that is, the actual length of road should be as short as possible. See Figure 5.1 for an example of cities connected by a network of roads. Notice that this would be a poor road network in real life, as drivers are forced to go the long way around between certain cities. Nevertheless, for the purpose of this theoretical problem, that does not concern us. However, that network is not the best network for these cities. The optimal network is depicted in Figure 5.2. There is no network connecting these cities with less road. Notice the addition of 3 interchanges, the smaller circle, outside of the cities. These interchanges allow the road network to be shorter than if all interchanges had to be within the cities. The task of evolution is to determine the best place to put interchanges, and which cities and interchanges should be connected. The cost of each network is the length of the roads, with a large penalty assessed to networks that fail to connect all the cities. Evolution proceeds by favoring networks with lower costs, producing mutated copies of them. The model also uses crossover, combining two existing networks to produce a new one. The end result is the evolution of a solution that is typically not optimal, but that is better than anything achievable by random chance. Unlike EV or Weasel, Thomas's simulation does not measure fitness as the distance to some target. It measures fitness as the actual quality of a particular solution. This gives it a much more feasible claim to actually be modeling natural selection. However, the veracity of this claim is lost by various details of the simulation that were fine-tuned in order to help the simulation succeed. One example of teleological fine-tuning is in the number of interchanges. The model restricts the number of interchanges to be at least 2. However, it is still possible that a potential network has interchanges that are not actually connected to any other cities or interchanges. For all intents and purposes, this is the same as not having those interchanges in the first place. But by requiring every network to have at least 2 interchanges, solutions that use fewer interchanges are rarer. What purpose does this restriction serve? The best evolved solution is one that does not use any interchanges approximately 30% of the time. Finding this solution is not impressive, as it can be found by random chance. However, without the restriction on the number of possible interchanges, it is the best evolved solution 96% of the time. The restriction exists to push evolution away from the undesired solutions without interchanges toward the better solutions with interchanges. It is teleological. Another source is in the initialization of the genome. Evolutionary simulations typically begin with a completely random initialization, but Thomas's algorithm introduces a restriction, forcing the interchanges to be in a narrower portion of the space, as depicted in Figure 5.3. Why this portion? Thomas knew that interchanges would have to be in the center of the frame and restricted the initial randomness to ensure that this happened. Again, Thomas is using his knowledge of the solution in order to aid evolution. Other examples of teleological fine-tuning can be found in this model. However, from the examples given here, we can see that Thomas engaged in teleological fine-tuning. Natural selection does not restrict mutations in order to help reach a goal, but that is what Thomas did. He knew about the solution he was trying to find, and made decisions based on that teleological aim and his knowledge. It is teleology, not Darwinism, that accounts for this success. Avida. Our final example is Avida. Avida evolves computer programs. For the uninitiated, the concept of a computer program may seem like an impenetrable dark art. But in essence, the idea of a computer program is actually simple. It is a recipe, a sequence of instructions that the computer follows. When following a recipe, a cook goes through the steps following each step's instruction, possibly skipping or repeating steps as instructed. A computer is doing the same thing. Avida evolves instructions for a task that can be visualized as mixing paint colors. When you mix yellow and blue, you get green. If you mix yellow and red, you get orange. If you mix blue and red, you get purple. By combining and recombining colors, you can obtain a wide variety of different colors. In order to obtain a particular color, you need the recipe, the instructions for how to mix the colors to obtain the desired color. Avida does not actually work with colors, but with 32-bit binary numbers. There are over 4 billion such numbers, but for the most part Avida works with only 16 of them. Think of these 16 as being 16 different colors. Table 5.1 shows what happens when different numbers are mixed. To use the table, take the two numbers being mixed, and look up the row and column corresponding to those numbers. The cell where the row and column intersect is the new color. For example, if you mix color 7 and color 10, then you will get color 16. Note that mixing a color with itself will not produce the original color, but a new color. The table is not random or arbitrary, it does follow rules. The nature of those rules does not concern us. Given an unlimited supply of color 6 and color 4, can you obtain color 10? Figuring out what mixes to perform in order to obtain the desired color is like a puzzle. It is possible, but it is not obvious how to do this. One possible recipe is: combine colors 6 and 4 to obtain color 15. Combine colors 6 and 6 to obtain color 11. Combine colors 4 and 4 to obtain color 13. Combine colors 11 and 13 to obtain color 8. Combine colors 8 and 15 to obtain color 10. There are a number of other recipes for color 10, but none with less than 5 mixes. It is also the only color that requires this many mixes. Within the Avida simulation, programs are favored by natural selection depending on which colors they produce. Colors 1, 4, 6, and 16 are not rewarded because they are easily obtained. The rest of the colors are rewarded, allowing the program to have more offspring if it produces more colors. Where is the teleological fine-tuning in this simulation? The most important example is the reward given to the colors besides the one being sought out. How do you produce a difficult color? First, you must produce the simpler colors. Once you have managed to produce those, you can recombine them to produce more difficult colors. If you repeat this process enough times, you can obtain the most difficult colors. The design of Avida facilitates this by rewarding the gradual development of increasingly difficult colors. Without that facilitation, Avida never produces the difficult colors. Avida rewards the simpler tasks or colors precisely because that helps produce the intended target color. The authors of Avida do not disagree with this assessment. In The Evolutionary Origin of Complex Features, they wrote, "Some readers might suggest that we stack the deck by studying the evolution of a complex feature that could be built on simpler functions that were also useful. However, that is precisely what evolutionary theory requires, and indeed, our experiments showed that the complex feature never evolved when simpler functions were not rewarded." The authors defend their decision to build the complex features, colors, on simpler features, colors, that were independently useful. But they do not appeal to any evidence that this would be a property of the real world. Instead, they appeal to a teleological point: in order for evolution to work, it needs the simpler functions to be rewarded. They constructed the simulation in this way precisely because it needed to be that way in order to work. There are some other aspects of teleology in Avida. Avida was designed to have particular properties in order to aid evolution. That renders it teleological and, thus, not a counterexample to the claim that non-teleological simulations do not produce successful evolution. V. Conclusion. In each of these 4 examples, the simulation works only due to being intelligently designed. Success is not due to some efficacy of the Darwinian process, but rather it is due to teleological fine-tuning by the programmer in the creation of the simulation. The programmers made decisions, whether consciously or not, that drew upon their intelligence to produce the desired result. In other words, the decisions were made teleologically. The precise nature of the teleological fine-tuning differs. In some cases there is an explicit target, and evolution has access to information about how far away it is from that target. In other cases, aspects of the solution are pre-programmed into the simulation. In still other cases, the problem itself was carefully chosen to have properties amenable to evolution. Whatever the individual case, the simulations work because of the teleological fine-tuning by the programmer. That teleological fine-tuning means that it is not a model of Darwinian evolution, and not a counterexample to intelligent design's claim. All of this is to defend the primary claim of intelligent design, that intelligence is a prerequisite to design. If these simulations were in fact teleological, that claim would have been falsified. However, as inspection of these simulations shows that they are teleological, the primary claim of intelligent design has been vindicated. So long as there continues to be no counterexample presented which demonstrates the production of design without teleology, the evidence for intelligent design grows. Other claims of intelligent design can also be tested by computer simulations. For example, work has been done evaluating irreducible complexity, low-impact mutations, biologically realistic mutation rates, and realistic fitness landscapes. All of these experiments confirmed predictions made by intelligent design researchers. Intelligent design has passed the test of computer simulations. Its falsifiable predictions about computer simulations are accurate. But what about Darwinian evolution? Does it make falsifiable predictions about computer simulations? Is there a computer simulation counterexample that would prove Darwinian evolution false? Evolution fails in some environments. Avida with a low mutation rate fails to evolve many tasks, Tierra does not start a Cambrian explosion, and Stylus fails to evolve a character starting from junk. However, these failures do not constitute counterexamples, because Darwinists allow that evolution does not always work. The conditions have to be right. But this renders Darwinian evolution unfalsifiable by computer simulation. No failing computer simulation can show that evolution does not work, just that the conditions were not right. If there were a theory regarding the necessary conditions, that could be tested. But as it stands, there is no such theory, and thus no computer simulation can falsify Darwinian evolution. If Darwinian evolution were correct about which computer models would succeed and which would fail, this would be strong reason to believe it was correct about biology. However, it does not even offer any predictions about which computer models will succeed or fail, making it unfalsifiable. Without falsifiability, no computer model, successful or not, can provide support for Darwinian evolution. The theory is equally compatible with either success or failure. It does not risk being falsified by a computer model, and thus gains no support from them. Writing at the blog Panda's Thumb, intelligent design critic Richard B. Hopper wrote, "Research using computational models of evolution are a thorn in the side of intelligent design proponents." But this is not true. Darwinian evolution is unfalsifiable, no computer simulation counterexample could prove it incorrect. As such, it risks nothing and gains nothing from any examples that demonstrate any sort of evolution. In contrast, intelligent design is falsifiable and makes accurate predictions. It forbids the evolution of design without intelligence, and makes accurate predictions about the limitations of Darwinian processes. The truth is that computer simulations are a nail in Darwinism's coffin, and a powerful demonstration of the predictive power of intelligent design. 6. The difference it doesn't make, why the front-end loaded concept of design fails to explain the origin of biological information. Stephen C. Meyer. Summary. Insofar as theistic evolution has been formulated with enough specificity to qualify as an alternative to neo-Darwinism as a scientific model, theistic evolutionists have typically affirmed that God created the universe and designed the laws of nature, including their finely tuned features. But having done so, they think that the origin of life and the origin of new forms of life can be explained by secondary causes, which they equate with the laws of nature and evolutionary mechanisms such as natural selection and random mutation. Thus, their view either entails the claim that the initial conditions of matter at the beginning of the universe and the fine-tuned laws and constants of physics contained all the information necessary to produce life, or it entails the view that the random mutation and natural selection, or some similarly materialistic mechanism, added significant amounts of new information into the biosphere since the Big Bang. The problems with the latter view are shown in chapters 2 and 8. This chapter demonstrates scientifically that the former view cannot be correct either, despite the claims of certain theistic evolutionists who argue for a front-end loaded concept of design that they call teleological evolution. In this view, the information necessary to produce life was present from the beginning of the universe. This chapter contends that this front-end loaded concept of design is scientifically inadequate because it fails to recognize that the laws of nature do not describe information-generating processes. As I noted in the scientific and philosophical introduction to this volume, the term theistic evolution can mean different things to different people. Some theistic evolutionists affirm that God actively directs the evolutionary process by, for example, directing seemingly random mutations toward particular biological endpoints. On this view, God has actively created new organisms by directing mutational change to produce new forms of life. This view has the virtue for theists of being at least minimally compatible with Orthodox Jewish and/or Christian doctrines of creation, since it affirms that God is actively doing something to bring life into existence. On the other hand, this view contradicts the, scientifically, orthodox neo-Darwinian view of the evolutionary process as a purely purposeless, unguided, and undirected mechanism, a blind watchmaker, as Richard Dawkins has called it. Indeed, as George Gaylord Simpson, one of the architects of neo-Darwinism, wrote in The Meaning of Evolution, the theory implies that man is the result of a purposeless and natural process that did not have him in mind. Other theistic evolutionists see the evolutionary process, including both the origin and subsequent evolution of life, as a purely unguided and undirected process, just as orthodox neo-Darwinists do. These theistic evolutionists conceive of God's role as much more passive. They conceive of God as merely sustaining the laws of nature, which in turn allow life to emerge and develop as the result of otherwise undirected and unguided mechanisms such as mutation and natural selection. While this view comports nicely with scientific materialism and neo-Darwinism, it seems to contradict religiously orthodox views of the creation of life by denying that God played any active role in creation or that he even knew what the evolutionary process would ultimately produce. Perhaps in an attempt to split the horns of this dilemma, some theists who accept the adequacy of materialist explanations have proposed a new view, or at least a view with a new name. For example, Dennis O. Lamoureux, a professor of Science and Religion at St. Joseph's College, University of Alberta, advocates a position he calls teleological evolution, or evolutionary creation. He prefers the term evolutionary creation to theistic evolution because, as an evangelical Christian, he wants to emphasize his belief in creation by making the term evolutionary merely the modifier rather than the noun in the description of his position. For him, evolution refers merely to the method through which the Lord made the cosmos and living organisms. He prefers the term teleological evolution to theistic evolution because he wants to affirm, contra Simpson and other neo-Darwinists, that evolution is a planned and purpose-driven natural process. But what exactly is meant by this idea? And does it provide an adequate scientific explanation for the origin and development of life? And if so, does it do so with enough specificity to be distinguished from standard materialistic theories of evolution and reconciled with a traditional Judeo-Christian understanding of God as the creator of life? According to Lamoureux, the theory of evolutionary creation affirms that the creator established and maintains the laws of nature, including the mechanisms of a teleological evolution. Lamoureux provides several illustrations to convey what he has in mind. For example, he suggests that God organized the Big Bang so that the deck was stacked to produce life. He also likens God to an expert billiards player who can sink all the balls on the billiards table in one shot. He likens the precise arrangement of the balls and the billiards player clearing the table with one single shot to God's initial act of creativity in bringing the universe into being with a very precise arrangement of matter, or initial conditions. Just as the billiards player can clear the table with one shot, God can create everything, the universe as well as all forms of life, with an initial act of creativity in which he arranges matter just right at the beginning of the universe and then lets it unfold deterministically in accord with the laws that he also established in the beginning. Thus, God needs no additional shots, no further acts of creativity, to bring life into existence. Lamoureux also compares the process of biological evolution to embryological development, in which an organism develops deterministically from a fertilized egg through time in accord with the laws of nature. Thus, Lamoureux seems to have in mind a kind of front-end loaded view of intelligent design in which the initial conditions of the universe are arranged or designed in such a way that life will inevitably evolve without any additional input or activity of a designing intelligence. As he explains, design is evident in the finely tuned physical laws and initial conditions necessary for the evolution of the cosmos through the Big Bang, and design is also apparent in the biological processes necessary for life to evolve. Thus, in some places he also refers to his view as evolutionary intelligent design. Although he uses the term intelligent design to describe his own view, he objects to the contemporary theory of intelligent design, and indeed, to any argument that implies that a designing intelligence played a role in the origin or development of life after the universe itself first originated. According to Lamoureux, to invoke a specific instance of intelligent design or divine action after the initial creation of the universe would imply a violation of natural law by invoking the activity of a God of the gaps. See our refutation of the God of the gaps objection in Chapter 19. As he explains, intelligent design theory is a narrow view of design and claims that design is connected to miraculous interventions, i.e., God of the gaps miracles that introduce creatures and/or missing parts in the origin of living organisms. For example, parts of the cell like the flagellum are said to be irreducibly complex, and as a result, they could not have evolved through natural processes. Since this is the case, ID theory should be termed interventionistic design theory. Lamoureux objects to intelligent design as an explanation for specific features of biological systems or events in the history of life because he wants to confine God's creative activity to the very beginning of the universe, and biological theories of intelligent design, he thinks, imply that an intelligent agent, quite possibly God, may well have acted at discrete points in the history of life after the origin of the universe. 3 problems with the theory of evolutionary creation, or teleological evolution, and its critique of intelligent design. Having developed an argument for intelligent design as an explanation for the origin of the information necessary to produce the first life, an event that happened well after the beginning of the universe, it should come as no surprise that I disagree with Lamoureux's critique of the theory of intelligent design and his theory of evolutionary creation. I do so for 3 main reasons. First, I see no reason to assume that the designing intelligence responsible for life and the universe, whom I personally believe was God, necessarily confined his activity to the very beginning of the universe. He may or may not have done so. I agree with Lamoureux that the laws of nature were established, and are maintained, by God. I also agree that the fine-tuning of these laws and the initial conditions of the universe provides evidence of intelligent design. Nevertheless, I see no reason to assume that this fine-tuning is the only evidence of design in the natural world. Nor do I think that the cosmological fine-tuning accounts for everything we find in the biological world, see below. Of course, Lamoureux finds appealing the concept of a God who had the wisdom to arrange matter so exquisitely at the beginning of the universe as to make any future actions on his part unnecessary. Others like to think of God as more actively involved in the process of creation. They find it appealing to think of God acting like a great composer who first establishes a theme at the beginning of his work and then adds new variations to that original theme at episodic intervals thereafter. As a Christian, however, I affirm that God acted entirely freely, and was under no compulsion to act in a way that either appeals to or affirms our aesthetic sensibilities. So I think the question of when God acted should remain a matter for empirical investigation and should not be determined by our aesthetic or theological preferences one way or another. As Robert Boyle, 1627–1691, often argued, the job of the scientist, or what he called the natural philosopher, is not to assume beforehand what God must have done, but to study the world to find out what God actually has done. Second, unlike Lamoureux, I do not think that material processes and mechanisms of evolution are sufficient to account for the origin of living forms, whether the origin of the first life or the major innovations in body plan design, see Chapter 2, that appear during the history of life thereafter. Yet, Lamoureux's attempted harmonization of theistic belief with evolutionary theory presupposes the adequacy and/or creative power of established evolutionary processes and mechanisms that supposedly demonstrate the incredible self-assembling capacity of the natural world during the distant past. But what if the evolutionary mechanisms that Lamoureux extols do not have the creative power or self-assembly capacity long attributed to them, as many scientists now argue and as the preceding, and subsequent, chapters in the first section of this book show? Before we construct elaborate theological harmonizations of the doctrine of creation with neo-Darwinism and other materialistic evolutionary theories, shouldn't we make sure that these theories are true? I think so. And for reasons that I have explained in preceding chapters, I, and even many leading evolutionary biologists, now doubt that known or even postulated mechanisms of undirected evolutionary change have the creative power long attributed to them. In my book Signature in the Cell, DNA and the Evidence for Intelligent Design, for example, I show that no undirected chemical evolutionary mechanism provides an adequate explanation for the origin of the information necessary to produce the first life. Jim Tour makes a similar critique of chemical evolutionary theory in Chapter 4. I have also shown in Chapter 2 and elsewhere that neither the mutation selection mechanism nor any of the other leading proposed evolutionary mechanisms provide a sufficient explanation for the origin of the major innovations in biological form, in particular, the novel body plans, that arise during the history of life on Earth. For this reason, I dispute Denis Lamoureux's view that known evolutionary mechanisms are the main method through which the Lord made living organisms. Third, I think Lamoureux's front-end loaded view of design is scientifically problematic. I do so because of my own study of the problem of the origin of the first life and the critically related problem of the origin of biological information. In the remainder of this chapter, I would like to discuss this problem and show why Lamoureux's theory of evolutionary creation, with its front-end loaded view of design, is insufficient to account for the problem of the origin of biological information, as well as the closely related problem of the origin of life. The origin of biological information, the fundamental mystery. In 1953, James Watson and Francis Crick first discovered the structure of DNA. It was the beginning of a profound revolution in biology that led to the recognition of DNA as an information-bearing molecule. Composed of linear strings of 4 different chemicals, called nucleotides, DNA carries in the linear sequence of these nucleotides information necessary to build organisms. The nucleotides function as do the alphabetic characters of written language. In language, the arrangement of letters to make words and sentences carries information. Similarly, the sequence of nucleotides conveys instructions for how to make the molecules and molecular machines the cell needs. The nucleotide base sequences in DNA also contain information about when and where to use those molecules. The information-bearing properties of DNA can be compared to that of digital computer code. As I noted in Chapter 2, this similarity is acknowledged even by neo-Darwinian biologists such as Richard Dawkins, who has observed that the machine code of the genes is uncannily computer-like. And software developer Bill Gates sees the similarity even more strongly. DNA is like a computer program, but far, far more advanced than any software we've ever created. Leroy Hood, famous for his contributions to biotechnology, describes the information encoded in DNA as digital code. But if this is true, how did the information in DNA arise? As it turns out, this question is related to a long-standing mystery in biology, the question of the origin of the first life. Indeed, since Watson and Crick's discovery, scientists have increasingly come to understand the centrality of information to even the simplest living systems. DNA stores the assembly instructions for building the many crucial proteins and protein machines that service and maintain even the most primitive one-celled organisms. It follows that building a living cell in the first place requires assembly instructions stored in DNA or some equivalent molecule. As origin of life researcher Bernd Olaf Kuppers has explained, the problem of the origin of life is clearly basically equivalent to the problem of the origin of biological information. Understanding Lamoureux's view, pick your poison. Denis Lamoureux does not directly address the problem of the origin of the first life or the origin of the information necessary to produce it. He doesn't say which specific naturalistic theory of the origin of life, if any, he favors. Nevertheless, the metaphors he employs to convey how God creates, deck stacked at the beginning, God as cosmic billiards player, evolution as embryological development, etc., imply that deterministic laws caused life to self-organize or self-assemble from some highly configured, and therefore information-rich, set of initial conditions at the beginning of the universe. Nevertheless, he does not say whether he thinks that, a, all the information necessary to produce the first and subsequent forms of life was present in the initial conditions of the universe, or whether, b, the laws of nature added new information during the subsequent self-assembly process. In any case, both proposals are scientifically problematic. So let's consider each in turn, starting with the second. Are laws creative? Lamoureux, like other evolutionary creationists, sometimes speaks as though he thinks the physical laws of nature might be generating the new information necessary to produce new forms of life. For example, he refers to evolution as a planned and purpose-driven natural process, and affirms that humans evolved from pre-human ancestors, and over a period of time the image of God and human sin were gradually and mysteriously manifested. Since Lamoureux disavows specific acts of divine creation as illicit appeals to a God of the gaps, and since he affirms that humans, at least, acquire new attributes and characteristics during the evolutionary process, it might be that he thinks that the laws of nature are generating the new information necessary to produce living systems and their unique attributes. But do laws of nature generate information? There are good reasons to doubt this. To see why, imagine that a group of small radio-controlled helicopters hovers in tight formation over a football stadium, the Rose Bowl in Pasadena, California. From below, the helicopters appear to be spelling a message, "Go USC." At halftime, with the field cleared, each helicopter releases either a red or a gold paintball, one of the two University of Southern California colors. Gravity takes over, and the paintballs fall to the earth, splattering paint on the field after they hit the turf. Now on the field below, a somewhat messier but still legible message appears. It also spells, "Go USC." Did the law of gravity, or the force described by the law, produce this information? Clearly, it did not. The information that appeared on the field already existed in the arrangement of the helicopters above the stadium, in what physicists call the initial conditions. Neither the force of gravity, nor the law that describes it, caused the information on the field to self-organize. Instead, gravitational forces merely transmitted pre-existing information from the helicopter formation, the initial conditions, to the field below. In the same way, the laws of nature do not add new information to that which was present in specifically arranged configuration of matter, or initial conditions, at the beginning of the universe. Laws can transmit but not generate information, as our illustration makes clear. There is a deeper reason for this. Scientific laws describe, by definition, highly regular phenomena or structures, ones that possess what information theorists refer to as redundant order. On the other hand, the arrangements of matter in an information-rich text, including the genetic instructions on DNA, possess a high degree of complexity or aperiodicity, not redundant order. To illustrate the difference, compare the sequence ababababab to the sequence one small step for a man, one giant leap for mankind. The first sequence is repetitive and ordered, but not complex or informative. The second sequence is not ordered, in the sense of being repetitious, but it is complex and also informative. The second sequence is complex because its characters do not follow a rigidly repeating, law-bound pattern. It is also informative because, unlike a merely complex sequence such as **** and ***J and 90te, the particular arrangement of characters is highly exact or specified so as to perform a communication function. In any case, informative sequences have the qualitative feature of complexity, aperiodicity, and thus are qualitatively distinguishable from systems characterized by periodic order that natural laws describe or generate. To say that the processes that natural laws describe can generate functionally specified informational sequences is, therefore, essentially a contradiction in terms. Laws are the wrong kind of entity to generate the informational features of life. The claim also betrays a categorical confusion. Physical laws do not generate complex sequences, whether functionally specified or otherwise. They describe highly regular, repetitive, and periodic patterns. This is not to malign the laws of physics and chemistry. It's simply to describe what they do. And yet, some scientists claim that we must await the discovery of new natural laws to explain the origin of biological information. Manfred Eigen has argued that our task is to find an algorithm, a natural law, that leads to the origin of information. But there is another reason that we will not discover such a law. According to classical information theory, the amount of information present in a sequence is inversely proportional to the probability of the sequence occurring. Yet the regularities we refer to as laws describe highly deterministic or predictable relationships between conditions and events. Laws describe patterns in which the probability of each successive event, given the previous event and the action of the law, approaches unity. Yet information content mounts as improbabilities multiply. Information is conveyed whenever one event among an ensemble of possibilities, as opposed to a single necessity, is fixed. The greater the number of possibilities, the greater is the improbability of any one possibility occurring, and the more information is transmitted when a particular possibility is fixed, specified, or elected. If someone tells you that it is raining, he will have conveyed some meaningful information to you since it does not rain, or have to rain, every day. If, however, he also tells you that today the raindrops are falling down, rather than up, he will not have told you anything informative since, presumably, you already know that rain always falls down, by natural law. As Fred Dretzker has explained, as p, psi, the probability of a condition or state of affairs, approaches 1, the amount of information associated with the occurrence of psi goes to 0. In the limiting case when the probability of a condition or state of affairs is unity, p, psi, equals 1, no information is associated with, or generated by, the occurrence of psi. This is merely another way to say that no information is generated by the occurrence of events for which there are no possible alternatives. Natural laws as a category describe situations in which specific outcomes follow specific conditions with high probability or necessity. Yet information is maximized when just the opposite situation occurs, namely, when antecedent conditions allow many possible and improbable outcomes. Thus, to the extent that a sequence of symbols or events results from a predictable law-bound process, the information content of the sequence is limited or effaced by redundancy. Thus, since natural laws do not generate or describe complex informational sequences, they cannot be invoked to explain the origin of information, whether biological or otherwise. Does the configuration of matter at the beginning of the universe explain the origin of biological information? To be fair, Lamoureux mainly seems to have a different scenario in mind, one in which the information necessary to produce living systems is entirely present at the beginning of the universe. Indeed, taken at face value, each of the metaphors he uses to describe his front-end loaded view of evolutionary intelligent design emphasizes how the deck was stacked at the beginning. Recall, for example, that he likens the evolution of the universe and life from the initial arrangements of matter at the beginning of the universe to the process of embryological development. He affirms that everything necessary to produce an adult organism, including, presumably, the biological information, is already present in the early embryo. It simply unfolds through time in accord with the laws of nature. In his billiard ball example, the precise arrangement of the billiard balls and the precise shot of the billiards player constitute information-rich initial conditions. The movement of the balls in response to the precise shot occurs deterministically in accord with the laws of momentum exchange. Thus, Lamoureux's metaphors imply that the information necessary to produce the first, and subsequent, living forms is already present in arrangements of elementary particles just after the beginning of the universe. But is this view scientifically plausible? Was the information necessary to produce the first life present in the arrangement of elementary particles just after the beginning of the universe? For this to be true, there must be a law that can transmit whatever information was present in the configuration of elementary particles at the beginning of the universe across billions of years without degradation, and also some process that can utilize that information, or convert it into a medium that can be used to produce the first living cell. Since the first living cell would, at the very least, require genetic information, that raises two more precise and analytically tractable questions by which Lamoureux's proposal can be evaluated. Was the information necessary to produce a functional gene, information-rich DNA molecule, present in the arrangement of elementary particles just after the beginning of the universe? Is there a physical law that could use the information present in the arrangement of elementary particles just after the beginning of the universe to produce a functional gene? In both cases the answer is no. And there are important reasons why this is so. First, it turns out that even the biologically relevant chemical subunits of DNA themselves do not contain the information necessary for producing a functional gene or the functional information DNA contains. And if they do not contain such information, then the much more simple and less biologically relevant arrangements of elementary particles, or distributions of mass energy, present at the beginning of the universe certainly did not contain such information. Second, there is no law that describes how these subunits self-organize into functional genes. To see why, recall what we know about the structure of the DNA molecule. What the structure of DNA reveals about the inadequacy of self-organizational models of the origin of life. DNA depends on several chemical bonds, each of which is governed by laws of chemical attraction, see Figure 6.1. There are chemical bonds, for example, between the sugar and the phosphate molecules that form the two twisting backbones of the DNA molecule. There are bonds fixing individual, nucleotide, bases to the sugar-phosphate backbones on each side of the molecule. There are also hydrogen bonds stretching horizontally across the molecule between nucleotide bases, making so-called complementary pairs. These bonds, which hold two complementary copies of the DNA message text together, make replication of the genetic instructions possible, just as chemical laws allow ink to adhere to paper. However, there are no chemical bonds between the bases along the vertical axis in the center of the helix, any more than there are laws determining that an A must always follow a B in the written English language. Yet it is precisely along this axis of the DNA molecule that the genetic instructions in DNA are encoded. Further, just as magnetic letters can be combined and recombined in any way to form various sequences on a metal surface, so too can each of the four bases, A, T, G, and C, attach to any site on the DNA backbone with more or less equal facility, making all sequences equally probable, or improbable, given the laws of physics and chemistry. Indeed, there are no differential affinities between any of the four bases and the binding sites along the sugar-phosphate backbone. The same type of chemical bond, called an N-glycosidic bond, occurs between the base and the backbone regardless of which base attaches. All four bases are acceptable, none is preferred. As Bernd Olaf Kuppers has noted, a present-day understanding of the properties of nucleic acids indicates that all the combinatorially possible nucleotide patterns of a DNA are, from a chemical point of view, equivalent. This fact about the structure of DNA was first noticed in 1967 by the physical chemist Michael Polanyi. In a seminal article titled Life Transcending Physics and Chemistry, Polanyi showed first that the laws of physics and chemistry generally leave open, or indeterminate, a vast ensemble of possible configurations of matter, only very few of which could have any role in a functioning biological organism. Specifically, he noted that the chemical laws governing the assembly of the chemical subunits in the DNA molecule allow a vast array of possible arrangements of nucleotide bases, the chemical letters, in the genetic text. In other words, the chemical properties of the constituent parts of DNA, and the laws governing their arrangement, do not determine the specific sequencing of the bases in the genetic molecule. Yet the specific sequencing of the nucleotide bases in DNA constitutes precisely the feature of the DNA molecule, namely its functionally specified information, that origin of life biologists most need to explain. Self-organization and evolutionary creation. In my book Signature in the Cell, I have shown why the chemical indeterminacy of the DNA molecule has devastating implications for self-organizational models of the origin of the first life and the origin of genetic information. Typically, these scenarios suggest that the forces of chemical necessity, as described by physical and chemical law, make the origin of life, and the origin of the genetic information that it requires, inevitable. But if law-like processes of chemical attraction do not account for the specific sequencing of nucleotide bases that constitute the information in DNA, as shown above, then such processes cannot reasonably be invoked as the explanation for the origin of the information in DNA in the first place. It turns out that the information stored in RNA also defies explanation by self-organizing forces of chemical attraction. Indeed, for those who want to explain the origin of life as the result of self-organizing properties or natural laws intrinsic to the material constituents of living systems, these elementary facts of molecular biology have devastating implications. The most logical place to look for self-organizing chemical laws and properties to explain the origin of genetic information is in the constituent parts of the molecules carrying that information. But biochemistry and molecular biology make clear that law-like forces of attraction between the constituents in DNA, as well as RNA and protein, do not explain the sequence specificity of these large information-bearing biomolecules. To say otherwise is like saying that the law-like forces of chemical attraction governing ink on this page are responsible for the sequential arrangement of the letters that give this book meaning. What does this have to do with the adequacy of the front-end loaded concept of design favored by advocates of evolutionary creation and other theistic evolutionists? Quite a lot. Evolutionary creationists such as Denis Lamoureux seem to affirm that the laws of nature as established and maintained by God are sufficient to produce life from the initial configurations of matter at the beginning of the universe. Thus, the evolutionary creationist position, with its emphasis on the deterministic unfolding of the history of life in accord with pre-established conditions and laws of nature, entails a commitment to some form of self-organizational origin of life scenario. In Lamoureux's billiard ball illustration, for example, a law of nature, the conservation of momentum, ensures a transition from an initial arrangement of matter, the balls arranged on the table, to a subsequent state, in which all the balls are resting in the pockets of the billiard table. Indeed, all his illustrations of evolutionary creation suggest that there must be some law-governed process that ensures a deterministic transition from the initial arrangement of elementary particles present at the Big Bang to a subsequent arrangement of matter constituting a living cell. He does not actually specify whether the laws that affect this transition actually generate new information or just transmit information already present in the initial arrangement of matter in the universe. Either way, though, he seems to assume that some law-like deterministic process must generate a living organism, starting ultimately from the arrangement of elementary particles, or distribution of mass energy, present at the beginning of the universe. As such, Lamoureux's theory of evolutionary creation entails a kind of self-organizational theory of the origin of life. And, indeed, Lamoureux himself extols the self-assembling character of the natural world. Yet, self-organizational scenarios fail to account for the origin of genetic information for the simple reason that there are no self-organizing forces of attraction that can account for the sequence specificity of DNA and RNA bases, the carriers of genetic information in all known cells. Moreover, the irreducibility of genetic information to the chemistry of the DNA and RNA poses a particular difficulty for the front-end loaded evolutionary creationist view of Denis Lamoureux and others. Evolutionary creationists insist that God's direct, discrete, or special creative activity has played no role in the history of the universe since the initial moment of creation at the Big Bang. They imply, therefore, that the known laws of nature acting on, presumably information-rich, configurations of elementary particles were sufficient to organize matter into the information-rich structures we see today in living systems. Yet, if the chemical subunits of DNA lack the self-organizational properties necessary to produce the informational sequencing of DNA, it is difficult to see how far less specifically configured and less biologically specific elementary particles or distributions of mass energy, present just after the Big Bang, possessed the self-organizational capacity necessary to arrange themselves by natural law into fully functioning organisms. In other words, if the chemical subunits of DNA, nucleotide bases, sugars, and phosphates, each constituting highly specific and biologically relevant arrangements of atoms, do not contain the information necessary to produce a functional gene, then the far less specifically configured and less biologically relevant elementary particles present just after the beginning of the universe most certainly lack the information to do so as well. But if this is so, then the deck was not stacked from the beginning as Lamoureux and other evolutionary creationists or teleological evolutionists have argued. Moreover, if the laws of physics and chemistry merely transmit information but do not generate it, then some other sources of information must have arisen after the beginning of the universe in order to produce life. The difference it doesn't make. In Signature in the Cell, I argue that intelligent design provides the best explanation for the information necessary to produce the first living cell. In the process of making that case, I critique the adequacy of self-organizational theories of the origin of biological information. Of course, neither self-organization nor intelligent design exhausts the logical possibilities for explaining the origin of information. One could also invoke contingency or chance, of either the directed or the undirected variety. For example, many chemical evolutionary theorists have invoked chance, usually in conjunction with prebiotic natural selection, see Chapter 4, in an attempt to explain the origin of information necessary to produce the first life. In Signature in the Cell, I show why those theories fail as well. Neo-Darwinists also invoke random and undirected mutations to explain the origin of the new biological information necessary to produce new forms of life once the first life had originated. In Chapter 2, and elsewhere, I have shown why those theories fail. On the other hand, some theistic evolutionists, such as Gordon Mills, have suggested that mutations generate anatomical novelty but that they are directed by a guiding intelligence. Unfortunately, none of these approaches involving contingency, chance or choice, represent live options for Lamoureux, who wants to attribute the origin of life to deterministic laws acting on finely tuned initial conditions. If, for example, the teleological evolutionist wanted to avoid the information-theoretic difficulties discussed above by invoking undirected chance variations to help explain the origin of genetic information, his position would become indistinguishable from standard materialistic versions of evolutionary theory, whether biological or chemical, that combine chance and law-like processes. Moreover, any version of evolution driven by such undirected, chance variations would not qualify as a purposeful process, as Lamoureux describes teleological evolution. If, on the other hand, the teleological evolutionist invoked directed contingency, the active or intelligent guidance of events, then he would violate his own self-imposed injunction against invoking divine action as a cause during the history of life and would commit a God of the gaps fallacy. Thus, the teleological evolutionist faces a dilemma. Either he sticks with the empirically unsupportable and theoretically incoherent view that natural laws generate specified biological information, or he invokes some form of contingency to account for the origin of biological information. If he claims that undirected stochastic processes generate biological information, then he would undercut his claim to have formulated a novel purpose-driven theory of evolution. But claiming that directed contingency generates biological information would entail affirming an interventionist version of intelligent design that Lamoureux explicitly rejects. Thus, it's difficult to see how Lamoureux's proposal can be rescued, at least on terms acceptable to him. Conclusion. C.S. Peirce's maxim, for a difference to be a difference, it must make a difference, seems applicable here. Lamoureux purports to offer a novel theory of biological origins. Yet, his theory is indistinguishable from self-organizational theories of the origin of life that cannot, for good reasons, explain the origin of the information necessary to produce the first life. Nevertheless, if Lamoureux were to modify his theory to overcome the inherent limitations of such a deterministic, law-driven approach, any such alteration would make it indistinguishable from other established theoretical approaches that he rejects. It's also unclear what difference his teleological approach to evolution makes. If the finely tuned arrangements of matter, or mass energy, at the beginning of the universe lack the information necessary to produce a single functional gene or protein, then even if God produced those arrangements, something else must be responsible for the additional information necessary to produce a living cell. If that something cannot involve the activity of a purposeful designing agent after the Big Bang, as Lamoureux insists, then clearly the process that generates the first life from that set of initial conditions cannot be teleological. All of which makes it fair to ask, does Lamoureux's specifically teleological theory of evolution add anything to the explanation of biological origins that a self-organizational theory, or, were he to modify his theory, a more conventional neo-Darwinian or chemical evolutionary theory, does not? Regrettably, the answer to this question is no. Since Lamoureux is unwilling to specify any role for God beyond the causally necessary, but insufficient, role of establishing initial conditions and maintaining physical laws, teleological evolution seems to be little more than an empty phrase describing a theory that, in the end, reduces to a standard, and completely inadequate, materialistic mode of explanation for the origin of biological information.